Good morning, ma'am. Morning, Ramesh sir. Good morning, Sarsiza madam.
Hello. Hello. Hello, sir.
Hello. Are you talking to me?
Sir.
Yes,
Vice
sir.
Chancellor.
Yes, sir. Good morning,
Madam,
sir.
Hey, good morning. Good morning.
Vice వస్తుందా మేడం?
Yes, ma'am. Yes, sir. This is clear.
Yes. VC గారు జాయిన్ అవుతారు. హలో. హలో. హలో.
Sir, we can hear you. It's clear.
Yes. Okay. Okay.
Sir, Viray, sir, good morning.
Hello,
Rangapalli,
good
sir,
morning, madam. Good morning. Good morning. Very happy to join in this conference. Hello?
Good morning, sir.
Hello.
Good morning, sir.
Very good morning, ma. It was in mute. Now
Yeah,
it's okay.
yeah. Now it is
This is-
clear, sir.
Okay. Good morning, sir.
Very good morning. Into offering. Automatic అదే పోతుందా?
మేడం మేడం
వీటిలో తగ్గిస్తున్నారు ఎట్లా వస్తాయి?
Sarsiza Madam.
Sir,
Madam, you can take initiation to start.
ఏంటంట ఏదో అంటున్నారు?
Madam, మీరు స్టార్ట్ చేసేయండి.
Yes, sir. I'm starting, sir. Murthy, sir.
Okay, madam. Now, thank you for the opportunity, my madam. Good morning to all the participants and dignitaries of today's international webinar. The University College of Science, Satavahana University, Karimnagar, Telanagana, India, organizing an international webinar on COVID-19 and expert analysis on variants, waves by an eminent vaccine scientist, Dr. Mahendra B. Deval Garu. And at this juncture, I would like to invite our today's patron and chief guest, our Honorable Vice Chancellor, sir, of Satavahana University, Karimnagar, Professor Mallesham, sir. Sir, good morning, sir. I would like to request to accept our virtual bouquet, floral bouquet, sir.
Thank you.
Madam, for the virtual bouquet, floral bouquet.
Say thank you.
Thank you, sir, for accepting our virtual floral bouquet.
Thank you very much.
Webinar, sir. And our chief guest of today's international webinar, our registrar, sir, Professor T. Bharat, sir. Sir, I would like to invite you and I request you to kindly accept our floral bouquet, virtual floral bouquet, sir. Professor Bharat, sir. Thank you, sir, Professor Bharat, sir, for accepting our virtual floral bouquet for today's national webinar. Thank you, sir. And I would like to invite today's speaker, the eminent vaccine scientist of Massachusetts, Dr. Mahendra B. Deval Garu. Sir, kindly accept our virtual floral bouquet, sir, for today's national webinar.
Thank you. Thank you so much, Narasimhamurthy sir.
Madam, kindly share the virtual bouquet.
మనం చేయలేము సార్ స్లైడ్ షో ఇది తీసేస్తాను.
Ma'am, thank you, sir, for accepting our virtual floral bouquet.
Thank you.
Dr.
Thank you so much.
Deval Garu. Thank you, sir. And the next, the today's guest of honor, the controller of examinations, Satavahana University, Dr. N.V. Rangaprasad, sir. So, Dr. Rangaprasad, sir, kindly accept our virtual floral bouquet, sir.
Thank you. Thank you very much.
For today's international webinar on vaccines and how to tackle COVID.
Yeah, thank you, Murthy. Thank you, Murthy.
Please, sir, Madam Chair. Thank you, sir, for accepting
Thank you.
our
Thank you.
reception as in the form of virtual floral bouquet.
Thank you.
Thank you, sir. And our another guest of honor, the OSD to Vice Chancellor, sir, Satavahana University, Dr. Vannala Ramesh, sir. Sir, I would like to request you to kindly accept our virtual floral bouquet, sir. Dr. Vannala Ramesh, sir.
Thank you, Murthy. Thank you.
Ma'am.
Murthy, thank you very much. Thank you.
Thank you, sir. Thank you for accepting our virtual floral bouquet, sir. Thank you, sir. And we are it's a time for the prayer, ma'am. According to our schedule, we have the time for the prayer. The prayer for commencement of our international webinar. At this juncture, I would like to hand over the remaining duty to our Honorable Principal, University College of Science, Satavahana University, Dr. Sarsaja Madam. She is the president of
Thank you,
today's
Murthy.
international webinar. Thank you, Madam. Please continue.
Thank you, Murthy.
Thank you.
A very good morning to all. This is Dr. M. Sarasija, Assistant Professor of Chemistry and Principal, University Science College, Satavahana University, Karimnagar, Telangana State. On behalf of University College of Science and my own behalf, I extend a warm welcome to the most respected chief guest of the webinar, Professor S. Mallesh Garu, Honorable Vice Chancellor, Satavahana University. Thank you, sir, for accepting our invitation in spite of your busy schedule. A warm welcome to distinguished special guest, Professor T. Bharat Garu, Registrar, Satavahana University. Thank you very much, sir, for accepting the invitation. I extend a warm welcome to renowned scientist and today's speaker, Dr. Mahinder B. Diwal Garu. Thank you very much for accepting our invitation to deliver an invited talk in the webinar. I welcome guest of honor, Dr. Bannala Ramesh Garu, OSD to Vice Chancellor, Satavahana University. Thank you very much, sir, for accepting our invitation. I welcome guest of honor, Dr. N.V. Sriringa Prasad Garu, Controller of Examinations, Satavahana University. Thank you very much, sir, for accepting our invitation. I welcome esteemed faculty members of Satavahana University and other universities and colleges. Thank you for your participation in the webinar. I welcome all the delegates, scientists, guests, research scholars, and students from India and abroad. Thank you, one and all. I would like to give a brief note about our today's webinar. Many people have misconception about vaccine, coronavirus variants, and third wave. Even today, some of the people are not willing to take vaccines. After lockdown, that is lifting the lockdown, many of the people are not following COVID-19 precautions, that is not wearing the mask, not maintaining social distance, not sanitizing effectively, etc. Many people are very much worried about third wave and its effects. In order to create awareness among the people about coronavirus, vaccines, coronavirus variants, and third wave, University College of Science, Satavahana University came forward to organize an international webinar. I am happy to inform you all that under the able guidance of Honorable Vice Chancellor, Professor S. Malesham Garu, today, University Science College, Satavahana University is organizing an international webinar on an expert analysis on COVID-19, coronavirus, variants, vaccines, and waves for the benefit of the people from India and abroad. Thank you, one and all. Now I would like to invite Dr. Namrata to introduce our today's honorable chief guest, Professor S. Malesham, sir.
Madam, am I audible?
Yes, you are audible, ma'am.
Okay. Good morning, one and all. Once again, on behalf of University College of Science, and I welcome you all to this colorful platform for this one-day international webinar organized by our principal madam, Sarasija madam, on the topic coronavirus variants, vaccines and waves, which correctly suits in this present situation where still we are pondering, swimming in this panic and confusion environment, how the third wave is going to progress and affect us. Now, in this occasion, it's my pleasure to introduce our Honorable Mallish Garu, Honorable Vice Chancellor of Satavahana University, Karimnagar. Professor Mallish sir is a renowned academician and a naval administrator who served as Vice Chairman of Telangana State Council of Higher Education during 2014 to 2017. Presently, he has taken charge at our Satavahana University as Vice Chancellor just 2 months back. And with this short duration, he is known to be an active, a flexible, and a perfect man with a lot of concern and cordial in receiving people and also in understanding people and also in all issues. Regarding Sir's academic background, Sir has completed his MA in philosophy with distinction and got his MPhil and PhD from Usmania University. Later on, with DAD fellowship, he went to Germany for research at Free University Berlin, and his research supervisors were one of the outstanding philosophers, Jürgen Habermas and Professor Axel Hannay. Sir has got 40 research publications presented at various national and international conferences. And during his long 29 years of experience, he served as head of the department, Usmania University, and as a principal, University College of Arts and Social Sciences during 2014 to 2017. And his main research contributions, his main research work focuses on the Nietzsche, is a German philosopher who is an outstanding philosopher for his ideas and which correlates with that of Dr. Ambedkar's philosophy. So what this Sir's philosophy reflects mainly is everyone must have a philosophy of life. That is, what purpose we were born and what is the purpose, what conduct, what behavior and what attitude we have to develop simply for achieving an academic, ethical, responsible and a peaceful well-being. With this short introduction, I wholeheartedly welcome Professor Mallish sir to this colorful platform for his valuable suggestions. Thank you one and all.
Thank you very much, madam, for a nice introduction. Now I request our chief guest, Mallish sir, to please deliver his valuable message.
Very good morning to all. Am I audible to everyone?
Yes, sir.
Yeah. Very good morning to all, particularly Principal Science College. Principal, I've observed Science College is very active in organizing all academic program with a lot of concern. For that, I appreciate all of your team members and you also, madam. Very good morning and congratulations for organizing this needy, timely topic, which everyone needs to be educated and more of, pushing people to know more about the coronavirus and its effects and all that, which is a very dangerous trend. World has first time experienced this. So your contribution in educating the people on this issue through this seminar will go a long way. I appreciate and congratulate you. And also I welcome and good morning to Satavahana University Registrar, Controller, OSD, and experts from different parts of the world, and also all the participants who are participating in the debate. I congratulate once again principal and also appreciate your spirit and initiative in organizing this needy program. Yeah, I don't know much about the scientific aspect of that, but definitely socially it has a greater effect on the world, people everywhere, everyone in the world. So what it requires is more of more of bringing awareness among the people. 50% of the medicine, yes, it is a medicine. With a medicine, we can address this. But other 50% of the issue, which is related with bringing awareness among the people, that is the most essential issue right now. So for that, we must all think of in a different way. This is one of the way where we educate people. In turn, they will go to different parts of the world and educate, bring awareness among the people. How, what are the precautions to be taken to be out of this dangerous situation. So for all this, I once again congratulate you and I welcome all the participants from all over the world and from different parts of the country. I thank you very much for giving me this opportunity. Thank you.
Sir, thank you very much, sir, for your kind words on this webinar. Thank you very much for sparing your valuable time. Next, I would request Prasad to introduce the registrar, Professor Bharat, sir.
Is it audible, madam? Is it audible,
Prasad?
madam? Is it audible, madam?
Yes, yes.
Okay. Good morning, one and all. Welcome to the international webinar on experts analysis and COVID-19. It's my pleasure to introduce our special guest, Professor T. Bharat sir, Registrar, Satavahana University, Karimnagar. Professor Bharat sir did his MBA, LLM and PhD from Usman University, Hyderabad. He has got 26 years of experience in teaching and 4 years of industry experience. He has published several papers in national and international journals in overall the world. He has received Best Teacher Award in 2014. He is currently working as a Dean Faculty of Business Management and Dean College Development Council, Satavahana University. His research area interest is in advertising, research, quantitative methods, and marketing. Now, I would like to invite our beloved registrar, Professor T. Bharat sir, to address the gathering. Thank you.
Thank you, Prasad, for inviting, sir. I request Professor Bharat sir to please address the gathering. Bharat sir. Sir, Bharat sir. Okay, I think due to technical problems, sir is not available here. Next, we will go for. Now I request Dr. Murthy to introduce Dr. Vannala Ramesh sir, OSD to Vice Chancellor.
Thank you, madam, for providing this opportunity. It's a great pleasure to introduce our beloved Dr. Vannala Ramesh sir. Ramesh sir is working as assistant professor in Department of Sociology, Satavahana University, and since its inception, he holds MPhil and PhD degrees from Usman University, Hyderabad. He is actively associated with academic institutions, NGOs, and government sectors. He is involved in project studies which are related to urban and rural poverty and child labor, child education, and tribal development, and gender studies. And moreover, he is worked on interview schedules, prepared them, and guided field investigators for planning and monitoring. And these projects duly recognized by the World Bank, state and central governments in India. He has published 14 research papers and chapters and books and journals. He has attended national seminars and presented more than papers. He has the distinction of attending international seminars in Georgia and Hawaii. Further, he has attended orientations and other programs, and he has worked as a project assistant, research associate, and research officer for different projects. He had distinction to serve as a member of Central Advisory Board on Child Labor, Ministry of Labor and Employment, Government of India from 2005 to 2008. He extended his services as a program coordinator NSS Satavahana University from 2009 to 2014. As a senior researcher, he guides 4 scholars in the Department of Sociology, and presently he also worked as a controller of examination Satavahana University and academic coordinator, and presently is the OSD to the Vice Chancellor in Satavahana University. Thank you, sir, for accepting as one of the guest of honor for the today's international webinar. Thank you, sir.
Thank you, Murthy. Thank you very much. Is it audible, Murthy?
Audible, audible.
Yeah, it's a good morning to one and all. Honorable Vice Chancellor Garu, Professor Malli, Satavahana University, and Mahendra Deval who is participating from abroad, and my dear colleagues and science college staff and research scholars, and who are participating from various countries and in India. So at the outset, I congratulate the teachers of University College of Science, Satavahana University, for conducting most relevant webinar in the present pandemic situation. Before COVID, we may not have considered how close we stood, walked or sat near other people. We have all made sacrifices over the past 1 year to keep our family and society in safe. This collective efforts has made huge difference. While restrictions are being lifted in some places, COVID-19 is not gone yet. We must continue to protect ourselves and each other. We need to change our lifestyle. This is due to COVID. And one more thing is, due to COVID, we are suffering from last 1 year. And one more thing, totally in India, migrant workers, skilled labor migrated from their respective places. Due to that, total industry and hotel industry, like other industries, due to lockdown also partially undeveloped. So and so many people, they lost their livelihood also. So, and one more thing is this, we are expecting third wave also. So already we are in second wave. This second wave is in different states, different parts. Is this wave is different methods they are working. So according to that, we have to modify our lifestyle and livelihood, and we should protect ourselves and our family and society. And one more thing is education system in India also going change due to COVID-19. So all these aspects you have to outcome, and this will help to our society. Once again, I congratulate to convener Sarasija Madam and Science College and University, and thank you for giving me this opportunity, Madam. Thank you very much. Thank you.
Thank you very much, sir, for giving a nice message of a present situation, that is COVID-19. Whatever the facts we are faced, you have given very detailedly about the COVID-19 effect. Thank you very much, sir.
Thank you.
And next, I would like to invite Professor T. Bharat sir, Registrar Satavahana University, to give his message. Sir, Bharat sir. Bharat sir. Okay, I think some technical problem is there with sir. Now we will go for next. Now I would request Mr.
Hello.
Sir, please, sir. Sir, please, sir.
Evaru? Madam.
Sir, please give your message. on this webinar.
Madam, am I audible?
Yes, sir. Yes, sir. You are audible.
Okay, thank you. Good morning, all of you. At the outset, I would like to congratulate the organizers for organizing an important topic, current topic, that is experts analysis on COVID-19 and its coronavirus variants, vaccines and waves. These three important aspects of the virus. So at the first, the world is experiencing one of the greatest challenges of humanity related to health problem, that is corona pandemic in the 21st century. The spread of the endemic is very fast and wide. This is the biggest problem. And the advancement of science and technology of the world is not faced the challenge effectively. The virus is spreading very fast and wide. The effects are very dangerous. And knowledge available to the people is very good and scant. So in this time, and the spread of knowledge, that is the behavior of the variant of the vaccines and varieties of the coronavirus, and we are talking about different waves, that is first wave, second wave, third wave, etc. We do not know much about that. In this particular situation, I think the experts' opinion and analysis may enlighten the people to understand them and effectively mold themselves accordingly. Also, to mitigate the dangerous effects of the virus to the people and society. So I think this conference or video conference may be successful in spreading the knowledge of this COVID-19 to help the people to mitigate the problems of the pandemic-related problem. Thank you, one and all. Thank you. Okay, madam.
Sir, thank you very much for valuable message, sir.
Okay, thank you.
Now I would like to request Mr. Joseph.
Yeah, madam. Good morning, madam.
Yeah, introduce.
Okay, madam.
Sriranga Prasad, sir, Controller of Examinations.
I am audible, madam. Is it okay,
Yes,
madam?
yes. You are audible. You are audible.
Good morning, all. At the outset, it gives me immense pleasure to introduce about Dr. N.V. Sriranga Prasad, sir, present Controller of Examinations, Satavahana University, Karimnagar. Sir holds MBA and PhD degree in Business Management from Kakatiya University, with 18 years of teaching experience, including 8 years of administrative experience in the capacity of principal. He is currently working as Assistant Professor in Department of Business Management, Satavahana University, and is also Head in Department of Business Management, Satavahana University. Further, he is also holding the responsibility of Controller of Examinations, Satavahana University. Prior to joining Satavahana University, Sir has worked in various capacities in reputed institutions like Hyderabad Business School, Geetham University, and Engineering Staff College of India. He has served as Executive Editor of the Indian Management Researcher, a peer-reviewed National Journal of Hyderabad Business School, Geetham University. Sir has published several papers in journals of national and international repute. He participated in various seminars, workshops, and chaired technical sessions for national conferences. He has written 2 books related to management studies. This is about a brief biodata of Dr. N.V. Sriranga Prasad, sir. Now I invite Dr. Sriranga Prasad, sir, to address the participants of this webinar. Thank you, madam, for giving this opportunity.
Thank you, Joseph, for giving this nice introduction. Thanks a lot. Good morning, one and all. Honorable Vice Chancellor, sir, Professor Mallish, sir, and respected Registrar, sir, Professor T. Bharat, and OSD2 VC, Dr. Ramesh, and other colleagues, and also convener of this program, Dr. Sarsija, and other colleagues of the department, of the chemistry department. It's indeed a great pleasure for me to participate in this international webinar on this topic of expert analysis in COVID-19 and virus variants and vaccines and waves. Indeed, for the past few days, recent past, we are just listening to only these terms. And after looking at the title, I thought that I think an acronym has been framed for this thing, like V3, V cube W, I think. I think hereafter we need to call like V cube W, right? The virus, variant, and vaccines and waves. So day in and out, and we are being witnessing to only these things, and I don't know. I mean, how we need to face such a kind of peculiar situation. I mean, in fact, we are passing out through a different kind of situation where we have not seen. I mean, the current generation have not seen such kind of a troublesome period where it has to how they need to tackle the situation and how they need to face it. And what is more important in this kind of scenario is your information. Yeah, information is available across your fingertips about your virus or statistics and all the things. But what lacks is the genuinity and credibility of the analysis or the information that is possessed or available at our disposal. And even we as a normal human being, I mean, a normal layman, we witnessing so much of articles and listening to news debates and all things. But every news item or every item what we see gives a different dimension, a different phenomenon, and that what where we are lacking about the credibility and reliability of the analysis. And I think this kind of an academic venture of providing and bringing out an expert who is considered to be an expert in this particular domain and bringing to the what do you call nearer to the people and public and giving an analysis and explaining about what is about this, as I said, VQW, what is a vaccine, why vaccine, and why this virus and how the virus is changing and what we need to do and what need to perform, what is our role in this context of maintaining ourselves healthy, and what is this wave kind of phenomenon. And wave kind of phenomenon is a very unique kind of situation we are talking about. Every time first wave, second wave, third wave, how many waves you are going to face is also unknown for us. And in this way, as I said, it's very unaware. I mean, as the lack of proper information is leading us to confusion, and this confusion is leading to what improper usage of the things is also is more very what you call different thing. And we generally we used to talk about studying behavior of people, but now I think that time has come for us to study about the behavior of the what you call virus and behavior of the variants and mutations and behavior of the waves. And then this is what the research is happening in this pandemic situation. And I think a lot of disruption has happened among our lives because of this VQ and W. And as such, this lack of information is creating confusion, as I said, and disruption is leading to disasters for us. And how we need to overcome this kind of disaster situation, maybe I think what we require is a proper, appropriate, reliable analysis, and that too from an expert like what we have today, Mahender Dewal. I think this kind of an expert analysis will give a great wider exposure and also information across the public and across all the sections of the society, so that we will be able to make ourselves what you call conveniently, we can take ourselves safety precautions and measures and have a lot of information. What is required today is a lot of information. I think that suitable and appropriate information we can expect from this expert opinion. And I congratulate the department again for conducting this apt program today. And I hope that this program will go in long way success and get a lot of information for all of us. Thanks a lot for providing me this opportunity to speak.
So thank you very much for your nice message, sir. Really, it is a very appreciable message, whatever you have given, the facts faced by us. And thank you very much for sparing your valuable time.
Thank you, ma'am. Thank you.
Yeah. Next, I would like to request Jayanti madam to introduce today's invited talkie, Dr. Mahender B. Dewal, sir.
Good morning everyone. Madam, am I audible?
Yes, ma'am. Yes, ma'am.
Good morning everyone. It gives me immense pleasure to introduce our outstanding biomedical scientist, Dr. Mahender B. Dewal, sir. Dr. B. Mahender Dewal, who is the founding member, Head of Research and Product Development, Cambridge, United States of America. Sir has a huge profile He is the biomedical scientist with 20 years of interdisciplinary experience in number of research areas, including organic chemistry, antibody engineering, chemical biology, and expansion microscopy. He has published 16 peer-reviewed papers in high quality and well-reputed journals, including American Chemical Society and various other publications. His research was featured in research highlights of Nature Chemistry and reputed as hot article in Organic and Biomolecular Chemistry. Sir also presently holds 4 US and international patents. He has been the president of Wales State University Postdoctoral Association and served as an advisory board member and organizing committee member. He has also been the vice president of MIT Ventureships and recognized student group, providing MIT students with working experience. He has received Excellence in Research Award by the New York Academy of Sciences and Cancer Research Award. And he has received his bachelor's degree with chemistry and biology from the Telangana, Nizamabad, Usmania University. Then he has worked as a research scientist at Department of Chemistry, Massachusetts Institute of Technology, Cambridge. And as the COVID-19 pandemic hit in December 2019, he started working on developing antibody therapeutics for COVID-19 infections. Presently, these antibiotic therapeutics are in preclinical studies on mice and hamsters. Additionally, from the past 1 year, Dr. Deval has been actively engaged in giving international webinars and participating in many debates in Indian news channels to raise the COVID-19 awareness, such as viral infection mechanisms, precautionary measures, and diagnostic tests. Dr. Deval spoke about the challenges of post-COVID-19 pandemic, a 24-hour World Book of Records organized by Lead India Foundation in the memory of our former President of India, Dr. APJ Abdul Kalam. Dr. Deval's service on COVID-19 fight had recognized him as a COVID-19 warrior by together Karnataka State Ministry of Youth Empowerment and Sports and SAARC Nations International Human Rights Association. So with this huge and enormous profile, I wholeheartedly welcome you, sir, for this webinar. And we thank you for sparing your valuable time and accepting our invitation for this webinar. Thank you, one and all. Thank you, madam.
Thank you very much, Jayanti ma'am, for introducing sir. Now it's time to request Dr. Mahender B. Deval to deliver his valuable lecture. Mahender ji.
Good morning, everyone. Thank you so much for a nice introduction, Dr. Jayanti Garu. First of all, I would like to thank University College of Science, Satavahana University, Department of Chemistry, the principal of the college, Dr. Sarasija Garu, and Department of Chemistry and all the departments for the invitation. Thank you so much. And I also would like to thank the chief guest, the Vice Chancellor of Satavahana University, Professor Mallesham Shankasala Garu, special guest, Professor T. Bharat Garu, guest of honors, Dr. Vamala Ramesh Garu and Dr. Sriranga Prasad Garu. Thank you so much for gracing this event with your presence. Thank you so much. So without a further delay, let me share my presentation and we'll get going. So can somebody enable me to share my screen? I think I'm disabled to share the screen. Someone has to enable me.
Sir, you are the host now. You can share, sir. Thank you.
Okay, thank you. So somebody says my voice is not clear. Can anybody hear well with my voice?
It is audible.
Is my voice clear?
Yes, sir.
Audible, right? Okay. All right. And I hope everyone can see my screen.
Yes, sir.
Right?
Yes, sir. It is visible, sir.
So, all right. Thank you. Thank you so much. So today I'm going to speak about COVID-19, the coronavirus variants, vaccines, and waves. So I'm going to start talking about variants, different variants. What factors making these variants lethal? I'm going to focus on that, and then I'm going to talk about the vaccines, how these variants are influencing the efficiency of the vaccines. Then I'm going to go into the multiple factors that are influencing the coronavirus waves, the number of waves coming, and various different factors that are influencing these COVID-19 waves. So first, I want to talk about the variants. So according to the CDC, Center for Disease Control and Prevention, coronavirus variants are classified into 3 different groups based on their impact on different parameters such as their transmissibility, disease severity, vaccine efficiency, and medicines efficiency. For example, the first class is called variant of concern. Why they are called as a variant of concern is because we have evidence saying that they're increasing the transmissibility, that means infectivity, and they have increased the disease severity in certain number of persons, and also they have impacted the diagnostic methods, treatment options, and influenced the efficiency of the vaccines. That's why they are called the variants of concern. And the second group is called variant of interest. These are called variant of interest because we don't have any evidence that they influence these parameters, but we can predict based on their mutations. The mutations happening in those variants, we can predict that they may influence in future all these parameters such as increasing the transmissibility or disease severity or reducing the antibody neutralization. That means influencing the vaccine efficiency or medicine efficiency. So these are all called the variant of interest. And the third group is called the variant of high consequence. These variants are called high consequence because they are not immediately impactful to the society, but they may become either a variant of interest or a variant of concern. So today I'm going to focus mostly on the variant of concern. So here is the table that displayed, which is generated by World Health Organization. You can see that here there are 4 different variants that World Health Organization recognized as a variant of concern: alpha, beta, gamma, and delta. These are called World Health Organization labels: alpha, beta, gamma, and delta. 4 different variants. But if you see these 3 other columns, there are 3 different types of naming systems: Pango lineage, G-side, and next strain. All different. These are a little difficult, more scientific. So to keep it simple for the common people, they named these World Health Organization labels with the Roman alphabets: alpha, beta, gamma, and delta. And on the right side, you can see where and when they evolved these variants. So let's not go into details on these details are not that important, but let's get started talking about these variants. Before we get started talking about the variants of coronavirus, we need to look at the structural features of the coronavirus, how it looks. So here is a cartoon picture of coronavirus. If you look at this cartoon picture here, coronavirus has a large genome with about 30 kilodaltons, which is a positive sense mRNA, which is securely encapsulated by a protein called nucleocapsid protein. You can see that the red color wires are the mRNA and the genome of the coronavirus, and the blue color the blue color dots are the protein that encapsulating the securing the genome of this coronavirus. And in addition to that, there are 3 other proteins, two membrane proteins, which is one of the membrane protein, which is in the purple color, you can see that here, and an envelope protein. Those 2 proteins give a robust structural features to the coronavirus. And another protein, which is called spike protein, which is a very important protein, gives certain specific features, which is very important in the infection mechanism, infection of the coronavirus to the human cell, as well as developing the vaccines. So let's take a look at a little bit more details into the spike protein and the infection mechanism in here. So on the left side, if you look at this, there is a spike protein on the virus membrane, which interacts with a membrane protein of the human cell, which is called angiotensin converting enzyme receptor, which strongly binds to that receptor. On the right side, if you can see that the animation that I have put in here, which was developed by Andrea Thorn's lab from University of Hamburg, Germany. So nicely using these spike proteins. Spike protein is a trimeric protein. 3 spike proteins come together as a trimer, and they attaches nicely to lands on the angiotensin converting enzyme like an helicopter, and then they attach, they strongly bounce, and then once it bound, you can see that there is a lot of mechanism is going on here. The orange and yellow color proteins from the human cell helps the virus to go under some structural changes, and then virus nicely attaches to the human cell membrane and fuses into the human cell and releases the coronavirus genome into the human cell. This is how the coronavirus infects the human cell using the spike protein. So spike protein, here I am displaying various different crystal structures or cryo-electron microscopy structures of the spike protein. On the right top side here, you can see that this is a cryo-electron microscopy structure, trimeric form of the spike protein in a vertical view, and this is a horizontal view of the same trimeric form of the spike protein. On the bottom down here, you can see the crystal structure of the spike protein which bound to the ACE2 receptor on the human cell. And this is the vertical view, and this is the horizontal view. So let's look into much more details, a little bit more details in the spike protein. Here is a monomer crystal structure of the spike protein with various different parts of it. The spike protein is majorly divided into 2 subunits. First one, this whole part is called S1 subunit, and this whole part is called S2 subunit. In S2 subunit, there are 2 crucial regions, which is called FP, which is furin protease region, where it gets cut by the furin protease from the human cells. And then the other part is called C-terminal domain. But S1 subunit has a major and important features of the spike protein, which helps the spike protein to infect the human, which are N-terminal domain, one part, and the second most important part is called receptor binding domain, which is RBD. In this receptor binding domain, this small part, which is called receptor binding motif, which is very, very important, plays a very important role in infecting the human being, binding to the ACE2 receptor. Here it is the monomer that bound to the ACE2 receptor. You can see in the zoomed region, this receptor binding motif, these 2 parts, you see that the kind of nicely clicks to the ACE2 receptor, strongly binds, and then makes the virus like easy to infect the human cell. And here it is the 2D map of the spike protein. It is a 287 amino acid protein with all these domains and subunits. But we are going to look into this most important feature, which is a receptor binding domain. And you can see that the amino acid number 332, 33 to 527. These amino acids are very crucial when they are mutated, and the variants are becoming more and more lethal. How we're going to look into the upcoming slides. And also in this receptor binding domain, in the blue part, there is a small red part, you can see that, which is receptor binding motif. That's what I'm talking about here, which is between 438 and 506 amino acid residues. So the variants, right, the coronavirus variants started in March 2020. The first mutation happened was D614G. An amino acid, aspartic acid, at 614th position, which located in the junction between S1 and S2 subunits, here, right over here, which mutated into a glycine amino acid in sometime in March. And that rapidly spread all over the world. 90 to 95% of the infections caused due to this particular variant. And if you can take a look at this, this is a real-time data. If you look at this came from the genomic analysis from the people who infected with this SARS coronavirus variant. In January, there is no mutation, 0 G glycine mutation. In February, 0%. In March, it started. In 26% of the patients have this glycine mutation, but April, May, you can see it's keep on increasing. By June sometime, it's 100%, more than 95% of the patients had this D614G mutation, and it's stabilized in the virus mutate, and it's been seen in the current variants as well. And also another thing is this mutation was tested in the lab that this is rapidly spread to the human cells compared to the D614. The D614 mutation is rapidly spread to the human cells compared to the D614 mutation. So now let's take a look at the variants. As I mentioned, World Health Organization excuse me, made this variant of concern, 4 different variants, right? So alpha variant, what kind of mutations are happening, where they are happening, what kind of influence they are having in the transmission of disease severity. So this is the alpha variant, which started in December 2020. I mean, recognized in December 2020 in UK, but it started a couple of months before. So there are several mutations all over the spike protein, you can see, including the D614G, which started last year March, right, in the junction region. But there are several different mutations. But you can see that the crucial part, the receptor binding motif, right? I mentioned like the amino acids between 438 to 506. One mutation, which is N501Y. This is causing rapid transmission, more 50% more infectious than the wild type mutation, wild type virus. And fortunately, this is only one mutation happening in the receptor binding motif, and it doesn't have much more impact on the monoclonal antibody therapies, and it has very minor impact on the vaccine efficiency. Most of the vaccines are worked very well. Let's say if there is a vaccine 94% efficient, then probably 1% went down, but now still the vaccines are very well efficiently cleared out this variant. The second variant is a beta variant, which is started in this, recognized in December 2020 in South Africa. And this, if you look at this, there are a couple of new mutations in addition to the N501Y, which we have seen in the alpha variant. The 2 mutations, which is E484K and K417N. All these are in the receptor binding domain, but now there are 2 mutations in the receptor binding motif, which is a very crucial part, as we have seen in my previous slides, that binding to the S2 receptor, right? Again, this variant also 50% more infectious than our original D614G variant. It significantly impacted the monoclonal antibody therapies. That means monoclonal antibody therapies from Regeneron, Lamivimab, as well as Adalimumab, are not properly working against this variant. The treatment is not efficiently working against this variant, and there's a moderate impact on the vaccine efficiency has been noticed due to this variant. The third variant is a gamma variant, and here this is not very much different. There are a couple of extra mutations in the spike protein here and there, but if you look at the receptor binding domain, same mutations as we have seen in the beta variant, but only the difference is the K417N changed to K417T in this gamma variant. Other than that, it is pretty much similar to the beta variant, and this was recognized in January 2021 in Brazil. It is more 20% more infectious than the beta variant, and there is a significant. This variant also showed significant impact on the monoclonal antibody therapy, and a moderate impact on vaccine efficiency as well. Now it comes to the Delta variant, which is recognized in April, May, when we hit by the second wave in India, 2021. And now, very interestingly, you see this variant also carries N401Y, which came from the alpha, beta, and gamma, all variants, which means this is stabilized. And E484 is also mutated in beta gamma, but it was E484K in the other mutants, other variants, but it is Q. There's a small change in here. In addition to these 2 mutations, another one is called L452R, new mutation accumulated, that too in receptor binding motif, which is very crucial to bind to the S2 receptor. That's why this is even more infectious than the other variants. 60% increase in transmission, 60% more infectious than the other variants, and it has a potential impact on antibody therapeutics as well, but we do not know yet. No data is available, but there's a lot of research is going on the Delta variant, because compared to the other variants, this is fairly new variant. It's only a couple of months old, and it probably has a potential impact on the vaccine efficiency as well, the Delta variants. So variant of interest. So this is again the table from the World Health Organization. These are kind of, you can say sisters and brothers of the variants of concerns. Pretty much they carry similar mutations in the other parts of the spike protein, but not in the receptor binding domain and receptor binding motif. That's why these are not as transmissible as the other variants. And you can see these are labeled as epsilon, zeta, eta, and these P1, P2, P3 are from the Brazilian variant, which is a gamma variant. The kappa variant is from the delta. This is the predecessor of the kappa variant. So these are variants of interest. Another one, recently we hear a lot in the news, which is the delta plus variant, right? So as we have seen in the other receptor binding motif, N501 for Y, for E484Q, and L452R. These three we have seen in delta variant. Another additional mutation added to the delta variant, which is K417N, which we have seen in the beta variant, right? So now it accumulated 4 mutations in the receptor binding domain, in addition to the other mutations in the spike protein. So there's not a lot of data available, but this is now into the variant of interest, not the concern yet, because there is a potential that it may, because we can see that there's a lot of 4 mutations compared to the other variant. There are only 3 mutations in other variants in the receptor binding motif, but now we can see 4 mutations, which may have a high impact on transmissibility, as well as the treatment options like monoclonal antibodies, and also in the vaccine efficiency. But there's a lot of research already going on. We do not have yet data. So now we have discussed, so far I have discussed about the various different variants, right? How they influence the vaccines? Let's take a look at that. So this table here, it displays approximately 12 vaccines developed, rapidly developed all over the world from various different academic and government as well as industrial organizations, except these 3. Vaccine from Novavax, Innovabio Pharmaceuticals, and Zydus. Rest of all the 9 vaccine candidates are authorized for emergency use in various different parts of the world, various different countries in the world. In India, Bharat Biotech successfully developed a vaccine. This is the first indigenous vaccine. This is also authorized for emergency use in India. Oxford AstraZeneca vaccine, which we call as a Covishield, also authorized in India. The other one is Sputnik V vaccine from the Malaria Research Institute from Russia, has been authorized for the use in India. And recently, another one vaccine, which is Moderna, has been authorized for the emergency use in India as well. And the other thing is this Zydus vaccine, NCO vaccine. This is a 3-dose vaccine, which they applied for emergency use authorization. They have conducted phase 1, 2, 3 trials. Right now, the application is with the DCGI. So this may get approved, authorized anytime from now, for the use in India. So let's start with the first vaccine. This is the BNT162b2 Pfizer vaccine, which is developed together by BioNTech, Pfizer and Fosun Pharma together with NIH, in a government agency in the US. This is a 2-dose vaccine, approximately 30 micrograms of mRNA that encoding the spike protein is encapsulated into lipid nanoparticles, will be given as a vaccine. So the first, it's a 2-dose vaccine, and second dose will be given after 3 weeks after the first dose, which showed about 95% efficiency on the original variant, which is B614G variant, from phase 3 clinical trials in various different countries, USA, Germany, South Africa, Turkey, and Argentina, approximately 44,000 participants. And also, can you please mute someone? Thank you. Okay. And also recently it has been authorized for the children, adolescent children between age groups between 12 to 15 years in the United States. And because we have a very nice data available from phase 3 clinical trials, approximately 3,000 participants in children and it showed 100% efficiency in the children. And the immune response retained up to 8 months. The antibodies developed due to this vaccination, memory cells developed due to this vaccination are retained at least 8 months, even more than that. But so far we have a data up to 8 months. Right now the clinical trials are ongoing in children, in kids, small babies, 6 months to 11-year-old children. but it has to yet to come. Hopefully, in the next couple of months, we may have this data, and then we can get started vaccinating children as well. And we have a strong evidence that this has a strong immunogenicity, which is in the pregnant and the women and the lactating mothers, which this evidence also showed that the antibodies developed by this vaccine also transmitting to the babies in the pregnant women as well as lactating mothers. So when this vaccine was screened against or tested against the new variants, alpha, beta, gamma, and delta, the efficiency went down. Particularly for the alpha variant, it's not that much. So like as I said, very mild impact on the alpha. This vaccine, the alpha variant, only 95% went down, but beta variant showed some really reasonable and decent impact on the vaccine efficiency. It went down to 75%. Though any vaccine above 60% efficiency is still as good as 95% efficient vaccine. The gamma vaccine variant, we do not have much data, but still it's a low. Delta variant, it's about 88%, similar to the alpha variant. This vaccine is really working very well against the delta variant, which means like 2.5-fold reduction in the neutralization capacity of the antibodies that developed by this Moderna Pfizer vaccine. So we do not have any data for the delta plus variant. So in next couple of months, hopefully we'll get some data. And also, all this efficiency went down for the infections only, not to the hospitalizations or deaths. Serious disease, it still protects 96 to 100% from causing the serious disease or hospitalization of the patient or death of the patient. So still, this vaccine is efficient against all different variants. Next vaccine is the Moderna vaccine. This is the second vaccine. Pfizer vaccine is the first vaccine which has been authorized for the emergency use against the coronavirus, and Moderna is the second vaccine that has been authorized for the emergency use against the COVID-19. And the Moderna vaccine is also mRNA-based vaccine, which is called mRNA 1273, an mRNA that encoding the spike protein similar to the Pfizer's has been encapsulated into the lipid nanoparticles. These lipid nanoparticles are kind of modified by PEG polymers, polyethylene glycol polymers. This Moderna vaccine has a little extra capacity in the lipid nanoparticles. So this is also a two-dose vaccine. Approximately 100 micrograms of mRNA is encapsulated into the lipid nanoparticles. This vaccine, the second dose will be given 4 weeks after the first dose, and this vaccine also showed similar to the approximately 94% efficiency in the phase 3 clinical trials from 30,000 participants in the United States. Also, there is a very nice data in the children, adolescents. Has been proved that it's 100% efficient, approximately 4,000 participants. The emergency use authorization they applied to the FDA. So anytime it will be authorized for the use in 12 to 15-year-old adolescents. And good news is that this has been authorized by DCGI in India as well. So as soon as this has been approved, we can get our children vaccinated in India. And the immune response from this vaccine retained at least 6 months. I believe it retained more than 6 months, but now we have data for 6 months only. And clinical trials are ongoing in children from 6 months to 17 years, as well as pregnant women and lactating mothers as well. So this vaccine showed pretty much 100% efficiency against alpha variant for the infections, against the infections. This is after 2 doses. 96% efficiency for the beta variant, and the gamma variant also decent activity, only 3-fold reduction in the neutralization capacity of the antibodies. Delta variant, 2-fold reduction of the neutralization capacity of the antibodies generated by this vaccine. We don't know the data for the Delta variant, and again, the hospitalizations and deaths protected from this vaccine is 96 to 100%, similar to the Pfizer vaccine. And additionally, this Moderna, this company, together with National Institute of Health, a government agency in the United States, developing two new vaccines. This mRNA 1273.341 is specific for the beta variant. And there is another multivalent vaccine mRNA 1273.211, which works on the multiple variants, not only the single alpha, beta, gamma. It can work beta, alpha, beta, gamma, or multiple variants. So they are under development, and then they have got very nice results, preclinical results as well as phase 1 data. So right now, phase 1-2 studies are ongoing. Hopefully, in a couple of months, these two new vaccines from Moderna may become available to fight against these new variants, or developing new variants from these coronavirus waves and viruses. And another vaccine is this adenoviral vector-based vaccine developed by Johnson & Johnson, a sister company of Johnson & Johnson. They have used human adenovirus type 26, which is a replication incompetent virus. In this virus, the genes that are associated to the replication of the particular this adenovirus has been deleted, and a gene that is encoding the spike protein will be inserted into this virus. And basically what they're doing here is okay, so I can continue. And the adenoviruses, human adenoviruses, are the common cold viruses. These are not pathogenic or lethal viruses, so that's why they can be utilized in the therapeutics. So what this vaccine does is this takes the gene, the spike protein gene, and delivers into the human body, and then induces the immune response. Depending upon the various parts of the world, it showed about 57 to 72% efficiency in the phase 3 clinical trials, and the immune response retains approximately at least 8 months. And this is another advantage for this vaccine is it's a single dose vaccine. You don't have to take 2 doses. If you look at the data for the variants, it has a 74% efficiency against alpha variant, only 3.6-fold reduction of the neutralization capacity from the antibodies, but unfortunately, only 52% efficiency in South Africa. People from South Africa showed, which is alarming for a South African population. This type of viruses, this type of vaccines. Gamma variant from Brazil, 66% efficiency. Delta variant only 1.6-fold reduction. But there's some studies suggesting that we may need a booster dose. For against the Delta variant, if you take a second dose, it may protect much more better than the single dose. Some studies are suggesting that, even lots of studies are ongoing right now, because the Delta variant is only 2 months old. We do not know a lot of information about that. Delta plus variant, we still have no data. And this vaccine also protects approximately 73 to 82% from hospitalizations and deaths from all these different variants. Excuse me. The other vaccine is Oxford AstraZeneca vaccine. This vaccine is called Covishield, produced by Serum Institute of India in India. And this vaccine initially developed by Oxford University together with AstraZeneca and Aborda, US government agency. This is also a 2-dose vaccine, but the dosage is recently we have a lot of data available, and this is also adenovirus vector-based vaccine, similar to the Johnson, but the virus they have used is a chimpanzee adenovirus. See, the similar mechanism that Johnson & Johnson adenovirus does, this vaccine also used, but it is a chimpanzee adenovirus, not the human adenovirus. So 2-dose vaccine. So second dose can be taken between 4 to 12 weeks anytime. There's a lot of data available, but this vaccine efficiently works if you take the second dose between 4 to 12 weeks. And it showed about 70% efficiency from phase 3 clinical trials in thousands of participants. And right now, there is a new studies are ongoing using as a heterologous vaccines, such as, first dose is AstraZeneca, the Covishield, and second dose is an mRNA vaccine. First dose is a Covishield, and second dose is other vaccine. So for mRNA-based vaccines, some data is published already, saying that it is safe in inducing robust immune response. Not yet authorized from any FDA or DCGI, but some data already produced that these are safe. Mix and match vaccines taking with Covishield or mRNA-based vaccines is safe. And this vaccine also working fine against all the variants. If you look at this, alpha variant 75% efficiency, Beta variant, this vaccine is highly impacted by beta variant in South Africa. Yeah, so only 10% efficiency. That means this vaccine is not at all working in people in South Africa or beta variant. Very low efficiency for the Gamma variant, and Delta variant it's showing 60% efficiency, which is a good, and a little bit more reduction in the neutralization capacity of the antibodies, but still working fine. 60% efficiency is good. Delta plus, we do not have much data, and it's also protecting. Again, this is very important. Hospitalizations of the infected people or death of infected people is 100%, 92 to 100% protected by Covishield or Oxford AstraZeneca vaccine. And another vaccine is a Sputnik V vaccine, which is developed by the Gamaleya National Center, Russian Direct Investment Fund together. And this is also adenovirus vector-based vaccine. Interestingly, they have used two different adenoviruses, adenovirus type 26, adenovirus type 5, as two different doses. Both are human adenoviruses. And you may have already noticed that this adenovirus type 26 was used in Johnson & Johnson's vaccine as well. And this is another vaccine, another adenovirus these Gamaleya National Center used to develop this vaccine. This is also a 2-dose vaccine. First dose will be given one of these, and second dose will be given the other adenovirus vaccine after 4 weeks of the first dose. This one showed, from the data, it showed that 90% efficiency from 3 clinical trials in Russia, approximately 22,000 participants. And this vaccine also effective against various different variants. Alpha variant, it's same as the original virus. The efficiency is not impacted for this virus. 3.1-fold reduction in the neutralization capacity of the antibodies produced by this vaccine for beta variant, which is not bad, and 2.8-fold for the gamma variant. For the Delta variant, not a lot of data available, only 2.5-fold reduction in the antibody neutralization capacity, but according to the Reuters website, they said 90% efficiency showed against the Delta variant from this Gamaleya vaccine, Sputnik V. Delta plus, we do not have much data, and as well as do not have data against the hospitalizations as well as the deaths. Still, a lot of studies are ongoing. Now, the other vaccine is a Bharat Biotech vaccine. This is a first indigenous vaccine developed by Bharat Biotech in India, BBV152. This is developed using the inactivated virus. The virus is taken and it will be inactivated using the heat or certain chemicals. That's why it is called inactivated virus. And or kill the virus. The virus will be killed using various different methods, such as heat and different chemicals, such as they have used beta-propyl-lactone to kill the virus in the Bharat Biotech vaccine. And they also added a couple of adjuvants, such as immunoglobulins. This is a toll-like receptor agonist, which stimulates the immune system. And Algel, aluminum hydroxide gel, as an adjuvant as well, which showed 78% efficiency in phase 3 clinical trials in 26,000 participants. All these studies are done in India on Indians. So this is really very good results. Right now, clinical trials in children and pregnant women are ongoing. So hopefully the data will come soon, and then our children and pregnant women and mothers will be vaccinated soon. And this vaccine also efficiently worked against the different variants from the published data. Alpha variant, same as the original virus. Beta, 2.7-fold reduction in neutralization capacity. Gamma variant, only 1.9-fold reduction. Delta variant, 3-fold reduction in neutralization capacity. And Delta plus, we do not have much data, as well as the hospitalizations. We do not have a lot of data from this vaccine as well. And the other vaccine is protein subunit vaccine, which is a new vaccine using the protein, spike protein itself. So the spike protein, you extract it from the virus, and then you synthesize it in the laboratory and purify it and then utilize it as a vaccine. And Novavax is the company that developed this protein subunit based vaccine, which is called NVX 2373. Serum Institute of India is producing it in India in the name of Covovax. It has not yet approved or authorized in any parts of the country or any parts of the world, but hopefully soon we have got some very good results from the clinical trials, about 89% efficiency in phase 3 clinical trials from UK and various other parts of the world as well. So hopefully it will be authorized soon so that we can vaccinate more and more people to protect from these variants and the coronavirus infections. And this vaccine also, they have used another adjuvant called Matrix-M. This is a proprietary adjuvant for the Novavax, and lipid nanoparticles. This is also encapsulated. The spike protein synthesized in the lab, encapsulated into the lipid nanoparticles before it delivered into the body as an injection as a vaccine. This, we do not have a lot of studies. Fairly, it's a new vaccine. It's not yet authorized. So, alpha beta variants. Alpha variant, we got like 62 to 86% efficiency has been shown. Some data showed from the publications. Excuse me, and it showed less than 60% efficiency in the beta variant and different parts of the world. But in South Africa, again, it's only 51% efficiency showed, which is alarming for the populations in South Africa, which they may not be able to use this vaccine for their people. And unfortunately, we do not have any additional data on the Gamma, Delta, and Delta Plus, as well as hospitalizations and deaths. Excuse me. So, the most importantly, I have explained so far that various different vaccines, they have developed using various different methods. But why these variants are influencing the efficiency or antibody neutralization capacity of the vaccine? What could be happening? Let's take a look at that. So, in general, when you're vaccinated, immune system generates two types of immune response. One is called humoral response, and second is called cellular response. Humoral response comes from the antibodies generated from the immune system, and the cellular response is and cellular response comes from the memory T cells and memory B cells that are generated by the immune system. So let's look into the humoral response, antibodies. So this is the spike protein. We are giving the spike protein as a vaccine into our body to stimulate the immune response, right? So immune system generates multiple different types of antibodies against the spike protein, which are called polyclonal antibodies. If you look at that here, 3 groups of antibodies are produced here. Some antibodies targets the receptor binding domain. Excuse me. Receptor binding domain, as I mentioned in my previous slides, right, which plays a crucial role in infection. So these antibodies stops the virus to infect the new cell. And N-terminal domain antibodies, some antibodies are developed against specifically for this N-terminal domain, and S2 subunit antibodies. So that means they surround the entire spike protein. The antibodies, polyclonal antibodies produced by the immune system, surrounds the entire spike protein and arrests it so that it will be neutralized. That's what we were all called neutralizing antibodies. So number of different. And if you look at this right side, this picture, this is the crystal structure. This is the receptor binding domain. Now here you see there are 4 different classes of even, this region. Antibodies developed by the immune system, several polyclonal antibodies, and also there are different classes in here. How do these classes work? Let's take a look at here. In general, during the infection mechanism, before the infection, the spike protein, it is kind of a closed, it exists in a closed conformation. That means the receptor binding domain is kind of a closed inside, it's kind of sleeping position. When it starts to infect, it becomes an open conformation. You can see that here, right? It just opens up and then it goes and clicks to the ACE2 receptor. That's how it infects the human cell and then releases the RNA into the human cell. But some antibodies, these are the crystal structures, real crystal structures. So, how they were created is the antibodies produced from the convalescent patients, they were extracted, isolated, and purified, then attached, they mixed with the spike protein and crystallized them. So, to see where they can bind. So, you can see that this particular antibody, C144, developed in the convalescent patient, they bind to the closed conformation of the spike protein. That means it may not open and it can never infect the human cell. It can never attach to the ACE2 receptor and it can never infect the human cell. And then the virus will be neutralized by the antibodies. Then the other cellular response for like cytotoxic T cells, macrophages, various different cells comes and clears out the major killer cells, clears out this virus from the body. This is how some of the antibodies work, number one. And you can, this is a much more zooming view, like, right, receptor binding domain, how they interact, this antibody interact to those amino acids in the receptor binding domain. That is crucial to bind to the ACE2 receptor. This is another antibody which is called B38. This was also extracted from the convalescent patients. It binds to this green, blue, and yellow. So this blue part binds to the blue here, and green part binds to the green. This is a receptor binding domain of the spike protein. And this purple part is the receptor binding domain here, right? So it interaction is very crucial here. This is a K417. You may have noticed that in one of the variants, right? This amino acid is playing a crucial role in binding to the human antibodies. That means this antibody may not work on one of the variants, which has this K417 mutation. Variants that has this K417 mutation, this antibody may not work. And importantly, this antibody B38, I have synthesized in my lab a couple of months ago. I have tested it against the pseudovirus, and it really works, really amazingly works against the pseudovirus, but it may not work efficiently on the variants that are. This is how antibodies. So when immune system produces multiple polyclonal antibodies, when virus changes its amino acids in that particular region, so some of the antibodies may not work. Let's say 100 antibodies, polyclonal antibodies are developed by the immune system. 3 or 4 may not work, which 3-4 amino acids are changed, right? Still 97% works. So that's how the efficiency goes down, the neutralization capacity goes down. If virus start accumulating those mutations more and more and more, the efficiency of the vaccine goes down and down, which takes more and more time, a longer time. But one important thing we need to keep in mind here is that the virus mutates when it has the host body. If it doesn't find a host body, it can never mutate. So if it finds the host body easily and rapidly transmits around, it can change its shape much more rapidly as well. So now let's discuss about the waves. So to discuss about the waves, I started with this slide, some statistics showing the infection rates from top 10 countries in the world. So you can see that as of today, approximately 190 million cases in the world detected with the COVID-19 infections, and 4 million people out of these 190 were dead. And United States with 34 million cases, number 1 position. India used to be 5th position before the second wave. Now it rapidly climbed up to the 2nd position with 31 million. Hopefully it stays there. And luckily, the death rate is lower than the Brazil. You can see that the Brazil is more death rate than the India, and other parts of the country, other parts of the world also impacted by severely. So I'm going to give some examples in here. So the best example is Israel. So the data, it's just, I did a crude analysis. This is not any mathematical model I have done. Rough and crude analysis based on the number of cases, number of daily new cases in each country. This is a rolling average of a week, 7 days, new cases, and deaths, hospitalizations, and vaccinations. So the comparative analysis I've done. So if you look at Israel, 2 waves. The first wave hit as rest of the world hit, sometime in August 2020, and it went down in November naturally. Still, we don't have any vaccines at that time, right? So again, second wave, they hit by second wave. And the second wave was much more worse than the first wave. But as soon as the vaccines are available, they started vaccinating the people. Now you can see that it completely went down to 0. Recently, it started spiking up. That could be because of the variants, slowly, which is probably the Delta variant mostly. However, if you look at this, okay, if you look at the hospitalizations, first wave, second wave rapidly increased, but almost nearly 0. It came down to 0, and recently it started going up, but it's not as rapid as the first and second waves. You can see that. And death rates, you can see first and second waves, rapid increase in the death rate, average new confirmed deaths, daily new confirmed deaths. Now it is almost zero. What could be the reason? Look at the vaccination capacity. This country, 60% of the population has been vaccinated at least one dose vaccine, and 57% of the population vaccinated, fully vaccinated with the 2 doses. This is a major reason. You can see that vaccinations are working properly to reduce the infections, deaths, and hospitalizations. But variants started increasing the infections and hospitalizations, but not the deaths. We need to keep it in mind here. This is what the data we can learn from the Israel. Let's look at another country, UK, right? So the first and second waves, as usual, the other countries, daily average cases, now it started hiking up again. You can see 40,000 in the country right now. It's going probably because of the Delta variant right now. Well, let's look at the hospitalizations here. It's not going as rapid as the number of cases went down, and deaths almost zero, nearly zero, 1 or 2 probably. Still, it's tightly controlled. The deaths and hospitalizations are tightly controlled. Why? Because of the vaccination. You can look at this. 60% or more than 60, about 69%. You can see that they may be 69% of the people vaccinated with at least 1 dose and 53% with fully complete 2 doses. They may reach to the herd immunity soon, probably in the next few months. Once they reach to 75%, they can get to the herd immunity, and then they may, even this can be also stabilized later. From this data, we can say that, again, vaccines are working very well to control the hospitalizations and deaths, but variants may cause in the spike. So again, probably, UK may hit to 3rd wave, but they are protected. Not a lot of people are going to die. Not a lot of people, hospitals are not going to flood with the patients. That is because the vaccination. USA. Now let's take a USA. Right, again, first, these 2 are the first waves, and this is the second wave. Very badly hit during the November, February, but as soon as the vaccine available in December, it went down. Now it started spiking up a little bit because the variants. But hospitalizations almost lower, very low hospitalizations. You can see that they're not spiking as the cases are going up. Deaths nearly zero, pretty much low, because you can see that 55.8% of the people vaccinated at least with 1 dose, but 48% fully vaccinated. So that means here also from the USA, what we can learn is variants may cause the spike in increase in the cases, infections, but the deaths, hospitalizations and deaths are controlled by the vaccinations, effective vaccination. Let's take a look at another country, which is Brazil, which is also badly hit by the COVID-19 pandemic. This is here it is the first wave. Number of cases went up and it came down. The second wave started. It's ongoing. It looks like it's going down, but you never know. It's going to go up. It's still the second wave is ongoing in Brazil. It's like a 2.0, 2.3, 3.0, 4.0. So second wave is still continuing. They're not even talking about the third wave. And we don't have much data on hospitalizations from Brazil. But deaths, they are going down. Death rate is significantly going down in the second wave, right? Why? Probably vaccinations. You can see that 43% people got vaccinated, at least with 1 dose. But only fully vaccinated people is only 15%, which is concerning. So let's take a look at a little bit more deeper look into what's going on in Brazil. Why second wave is ongoing? Why not the death rate is even coming down to 0 even though they're getting vaccinated? So when I looked at some literature, some information, I found this nice news article in the New York Times. So the very first thing that they noticed is vaccine scarcity. Vaccine scarcity is not because vaccines are not available for them. The governments, the administration decisions, basically. So they have done lots of clinical trials for various different vaccines, Johnson & Johnson vaccine, Oxford AstraZeneca vaccine, Sputnik vaccine, some of the Russian vaccines. They have done clinical trials in Brazil, but the administration was not rapidly acted to buy them to give it to people. That's one of the reasons, vaccine scarcity. Second thing is, people are not following the COVID adaptive behavior. You can see that as soon as the shopping malls are open, restaurants are open, beaches are open, people are not wearing the masks. They're not really much more concerned with the new infection. So that's another reason for the second wave not to stop. And the third reason is the new variants. Again, as I mentioned in my previous slides, Brazil has its own variant, which reinfecting, breakthrough infections. Like, people are, once they got the infection, they're getting infected again with the new Brazilian variant, P1 variant. Now it's called P2 variant. The new variant also started in Brazil. These are the reasons why Brazil is still continuing with their second wave. Let's look at South Africa. South Africa is one of the bad situations. The country hit very badly by the COVID-19 pandemic. First wave, second wave, already third wave ongoing right now in South Africa. Death rate is also ongoing, going all the way up. But you see, the vaccines are not going up. Only 7% of the country vaccinated, 2.9% fully vaccinated, which is not at all good. We can clearly see that vaccine influence, how vaccine influence to control the deaths and the hospitalization. We don't have the hospitalization data from South Africa, but the infections, deaths, and hospitalizations we have seen from Israel, UK, and US, right? But here the vaccination rapidly is not going on. And I have done another bit more research. So this article published in news article published in BBC News. So what kind of a problems vaccine South Africa experiencing? The most important thing is vaccine scarcity, because the problem with South Africa, they are not developing their own vaccines. They are relying on other countries for the vaccination, and mostly on India, the South Africa, and only less than 2% of the continent, South Africa has been fully vaccinated. We have seen it, right? That's scary because they don't have a vaccines available. The second thing is South Africa already had its own beta variant, which is highly lethal. You can see that, right? From the data of previous slides, you have seen that. And now they are hit by the Delta variant as well. So variants also causing the third wave continuation, third wave starting and third wave continuation in the South Africa. And let's take a look at Russia. Russia also started again. Third wave already started in Russia. This is a surprising data I have seen from Russia. First wave, second wave, third wave is ongoing, increasing the cases. We don't have the data for the hospitalizations, but the death rate is not at all going down. And the one interesting thing is Russia is one of the first countries authorized their own vaccine in August, September, sometime for Russians. But the vaccination speed is very slow, only 20%, 22% of the single dose and 14% with the 2 doses. What could be the reasons? You can clearly see that the vaccination influence on the third wave as well as the increase in deaths. The deaths are not going down at all. That's what is the biggest problem in Russia. So what could be the reason? Let's take a look at that. The major problem is vaccine hesitancy. If you look at that, Russians are not trusting their own vaccine. That's the biggest problem. So let's wait and see. We'll see, and then some others will take. If they're safe, we'll take it. The other thing is variants. Recently, the Delta variant entered Russia, and then that's also causing lots of chaotic situation. Going on third wave in Russia and increased deaths as well. So, from this data, this waves, this real-time data, the factors that I kind of summarized in the slide. What kind of factors that decrease the chances of COVID-19 waves, the COVID-19 wave? It could be first, second, third, or fourth, fifth, sixth wave, who knows? You can notice here is the increased vaccination from the countries like Israel, UK, and US. No vaccine hesitancy. There may be some vaccine hesitancy, and you can see probably that little spike in the cases due to this vaccine hesitancy. But heavy vaccine hesitancy in Russia and then spiking the cases as well as the deaths, you can see that. Concern toward the new variants. People need to be careful, very careful. Following the COVID adaptive behavior, even though you are fully vaccinated, they need to continuously follow the COVID adaptive behavior, such as wearing the mask, sanitizing the hands, and washing the hands, maintaining the social distancing, avoiding the social gatherings. See, these are really, really important, even though vaccinated, because we need to reach to the herd immunity. Until we reach the herd immunity, we still need to follow this behavior. These are the factors, same factors. Influenza increases the wave, right? Vaccine scarcity, example is South Africa. Hesitancy, vaccine hesitancy, best example Russia. Slow vaccination, best example Brazil. And new variants from all over the world, and even UK, the best example. Variants are causing increase in the number of cases, and people's careless attitude. not following the COVID-adaptive behavior. These are the main reasons to cause any second or third wave. So what is happening in India? Let's take a look at the real-time data in India. So we were very, very careful in the first wave time, right? Because we have learned from the world and then we imposed strict rules. So we control the deaths or infections or anything in the first wave. But as soon as we neglected the virus or pandemic, we hit by the second wave. And you can see that crazy increased number of cases. We don't have the hospitalization data. And because of, again, the regulations and then, strict rules and, lockdowns and all measures, the death rate went down. Now we're at the edge of third wave or second wave. Is the second wave is going to continue or third wave is going to come? Let's look at the vaccination, how fast we're going based on the population. So we can see that 23% is not quite sufficient for the population that we have, right? At least single one dose. 23% of the population received one dose only, and 6% of the population received fully 2 doses. But from Brazil, South Africa, and Russia, we can learn that increase and US, UK, and Israel. We learn that the increasing number of fully vaccinated and single dose of vaccination reduces the death and avoids the third wave. That's what we learned from the real world data from the other parts of the world, other parts of the world. But is that what is happening in India? Let's take a look at that. See, we can see these people, right? How people are, as soon as the lockdown is gone, and now you can see that. And we have the Delta variant ongoing. Now we are talking about the Delta plus variant is oncoming. Well, one good news is, 8.6 million doses were administered in one single day. That was sometime in this month or last month. That's a good news. But is it sufficient? We need to think about it. I think, is it slow? Do we need to speed it up? We need to reduce the vaccine hesitancy. We need to increase the vaccine awareness. So I think, after looking at this waves data, I think you have a better idea than me whether the third wave is going to come in India or are we going to, the third wave will be stopped in India. So you can tell me better than that, better than me probably. But in the summary, so as of now, I have discussed about the spike protein mutations of the coronavirus variants and how they're influencing the vaccine efficiency, such as and then I give you real-time examples on the COVID-19 waves in the world, how countries are fighting against the waves and variants with vaccines, and how vaccination protects from the waves as well as the variants, and not only the vaccination but also public's responsible attitude that is most important to avoid any wave or any increase in the search. Last but not least, what I would like to say is always, again, still, even though you are vaccinated, fully vaccinated, until we reach to the herd immunity, I would recommend to follow these non-pharmaceutical interventions, wearing the face mask, washing the hands, sanitizing the hands, practicing the social distancing, avoiding the social gatherings like parties, weddings, festivals. This is not happening in India. These are really very concerning. And the more important thing I always recommend people is boosting their immune system by eating nutritional food, getting sufficient sleep, being an active lifestyle, leading an active lifestyle, at least 30 minutes a day, either walking or running or some kind of physical activity. Having said that, in this my journey, there's a lot of people from my chemistry journey started from Department of Chemistry, OU campus. I started learning chemistry from there. Then I briefly worked at IICT India, where my first research career started in IICT India. Then I moved to University of South Carolina for my PhD. Then I moved to Wayne State University for my postdoctoral studies. And then I got my first job at Massachusetts Institute of Technology, MIT, in chemistry department as a scientist. Now, I was involved with a new startup company, which is Expansion Technologies. Right now, we're developing novel therapy, diagnostic methods for early stage detection diagnostic methods for various different cancers. As the pandemic hit, we started developing new antibody therapeutics against the COVID-19 as well. So in this my journey, many my mentors, my gurus, my colleagues, my friends, without their help, I don't think what I am today and where I am today. And last but not least, my family members and my best friends also involved in my journey. I would like to wholeheartedly thank each and every person helped me as to grow as a scientist. Thank you so much for your attention. My apologies if I went a little over the time, but I'm happy to take any questions you have.
Sir, it's very informative and knowledgeable session, sir. There are a few questions from the participants. So
Please go
on
ahead.
behalf of participants, I would like to ask, with your permission.
Please go ahead. Yes,
So
no
first
problem.
question is, so I think it has been already explained by you, but once again, the question has been raised by the participants. So I'm asking them, what is the effectiveness of Covaxin and Covishield on different variants in India?
In India, so we do not have the efficiency data, but there are some studies have been done. Like, for example, the serum from the vaccinated person are taken and tested against these variants. How effectively they are neutralizing the virus variants. In that study, for the Covaxin, in one of my slides, I mentioned, I think around 2 to 3-fold reduction in the neutralization capacity, which is negligible. It's not that bad. I mean, obviously, right, there is a mutations in the spike protein, particularly in the receptor binding motif, then obviously we would expect some of the antibodies may not work. But again, immune system develops when you get vaccinated. The immune system produces polyclonal antibodies, multiple antibodies that surround the spike protein, and there's some lock them in a closed conformation so that it will not infect to the human cells. Probably some of the antibodies that bind to the closed conformation or may not be actively working, so that it still becomes an open conformation and then probably infecting some of the patients. But again, still that's 1 or 2 cases, 1 out of maybe 1 lakh or 2 lakh. So still, again, Covishield, Covaxin, both of them are effectively working against all different variants, including the Delta in India.
Thank you, sir. There is one more question. That what is the efficacy of Novavax with regard to the biocompatibility and
Go ahead. Can you first introduce yourself? Who is asking the questions? I think you can ask questions later, but I think there are some hands raising I've seen. Professor D. Ashok wants to ask a question. Probably, sir, unmute yourself and ask the question, sir. We can go with the participants' questions once we have these interaction session with whoever wants to ask the direct questions.
Dr. Mahenderji.
Sir.
Dr. Mahenderji.
Yes,
Hello?
sir. Go ahead. I can hear you. Can you hear me?
Okay. So far, in any country, herd immunity is developed or not, or how much time required to develop herd immunity?
Good question, sir. So, I mean, the countries that are analyzed from the real data, so to get the herd immunity, you need to 75% people either have to be vaccinated or some part of the 75% already get the infection and recovered. So 75% people has to be developed sufficient immunity against the virus. Then only you can get this herd immunity point. But most of them, I haven't seen any countries. I think UAE, United Arab Emirates, kind of close to that herd immunity, but still, excuse me. So again, variants are creating some new cases for them as well. So as soon as the new variants come into the picture, they may cause some disturbance. But they control the deaths, and hospitalization. The vaccination only thing controls the deaths and hospitalizations. So how much time? It depends on how fast the countries vaccinate their people.
Okay.
And that's another thing. Second thing is, again, we don't. The problem with this problem is it's not the country. The whole world has to be vaccinated rapidly. Because even though, let's say, some small country in South Africa has remained not vaccinated well, then the virus may get back again and then go back and circulate around the world. So that's why World Health Organization, the other US, Australia, UK, European Union, they're all trying to produce more vaccines and supply to the other parts of the world, so that we can overcome this pandemic rapidly. But sir, to be honest, several months are going to be taken. We still need to live careful life for several more months. I mean, we had a chance before the second wave hit in India. We had an opportunity. If we lived like another few months carefully with our COVID appropriate behavior, we could have overcome. But I mean, in my slide, still people seems like they haven't learned.
I have one more question.
Sir, go ahead, please.
There are different types of vaccines.
Yes.
The first dose, second dose must be same. In case same type of vaccines can be given as first dose and as second dose, two different of similar
Excellent
type of
question,
vaccines.
Sir. It's exactly. It's an excellent question. So we do not have the much data. So theoretically, I can answer your question theory. Right?
Okay.
We have one example. One example we already have, which is the Sputnik Russian vaccine. Two different adenoviral vectors are used. One adenovirus type 5, second dose is adenovirus type 26. And now they're talking about the Sputnik V Lite, which is nothing but adenovirus type 26, which is mostly similar to the J&J vaccine, Johnson & Johnson vaccine. They are also using adenovirus type 26 to develop the vaccine. So now we have an example. So that means probably we can adenoviruses can be mix and match. Like Covishield is an adenovirus vector vaccine, but that's a chimpanzee adenovirus vector. So again, another Chinese company is called Consino Biologics. They developed another vaccine, which is another adenoviral vector-based vaccine, which is they have used type 5 adenovirus human adenovirus vector, which is already used in Russian vaccine. So there is a possibility. Theoretically, you can see that. I mean, if I have to take it, I would take it, but I cannot recommend to someone unless until I see a data.
Okay.
But theoretically, if you are comfortable, confident, you may be able to.
Yes.
And other thing is Covaxin is a inactivated virus-based vaccine. So if another inactivated virus-based vaccine, there's a possibility, not a problem. I don't see any problem. But the other thing is, even though if you let's say you take the vaccine, either any type of a vaccine, right? The immune system will be rapidly activated within 2 weeks, 2 to 3 weeks, maximum 4 weeks.
Yes.
It will be activated, developed, all the antibodies are ready, memory cells are developed, they're there.
Yes.
And if there is any residual vaccine or any residual parts of the vaccine will be cleared out by body within weeks. So we do not have to worry about that.
Okay.
The only thing we need to worry about it is how rapid immune response is going to be developed. Let's say when you mix and match. So for example, some data available for the Covishield and mRNA vaccine from the from European Union. So some participants were given first Covishield, and then after 4 weeks they were given mRNA vaccine.
Okay.
People for above 60 age group, they got a little bit more rapid side effects. Rapid side effects means fever was severe fever, normal side effects, not the longer term side effects. Side effects means rapid response because the immune response was much faster, quicker. So they experienced a little bit extra pain on the hand, and then severe fever, severe little bit of a nausea kind of things they experienced. But below 60 year age group people, they were fine. So the data available now for the Covishield and the mRNA vaccines, but not yet authorized by the government agencies. So we still have to wait to recommend for people.
Okay. Thank you.
Maybe a long answer.
Yes, sir. Thank you.
I think
Dr.
G.K.
Ramana would like to ask.
Yeah, please ask. Unmute yourself and ask G.K. Ramnagar, please. If he's not there now, I think Preeta Ghosh, you can unmute yourself. And is G.K. Ramanagar still there?
Yes, sir. Sir?
Yeah, go ahead, Preeta. Yeah.
Yeah, sir, very good morning, sir. This is my, I heard your previous lecture, which was held at St. Pius College, Department of Chemistry organized. Uh, Thank
That
you
was
very
last
much,
year.
sir. Yeah, that was last year. So I do have 3 questions with me, sir.
Please go ahead.
The thing is, as you said that Novavax, it is a nanotechnology based, the vaccine production is based on nanotechnology. So basically with regard to the efficacy concerns, when it comes to the biocompatibility and toxicity issues, so what is the level of protection it offers to an individual, the candidates?
Okay. Let me answer this first. So, so nanotechnology, it's not the nanotechnology. I mean, well, it's sort of a nanotechnology-based vaccine. So lipid nanoparticles, yes, it's a nanotechnology particle. So lipid nanoparticles, the spike protein, pre-fusion conformation of the spike protein will be synthesized in the lab and purified, and then it will be encapsulated into the lipid nanoparticles. This is a method very well established for the toxicity studies. So lipid nanoparticles are being used to deliver not only the vaccines, several different types of medicines, medications, and various different diseases. This technology has been there for several decades. So the toxicity studies have been vigorously done, animals, humans, various stages of clinical trials. So we do not have to worry about the toxicity because we have a sufficient data that they are very safe, number one. Right. Number 2 is the delivery method. So these lipid nanoparticles also, they are kind of various different methods to develop these lipid nanoparticles, and every company has their proprietary technology, right? So that depends on how efficiently it's going to deliver to the into the body. The rapid immune response depends on how efficiently it will be delivered. So to my knowledge, Novavax got 89% efficiency. That means it's doing pretty good, right? It's a rapid response. So I wouldn't worry about that the toxicity at all. I mean, even the 30,000-40,000 people participated in the phase 3 clinical trials. One of my friends participated in the clinical trial of Novavax clinical trial. I mean, he said he didn't see anything. So that vaccine is safe and effective as well. What is your other question?
Yeah, the other question, sir, is there any possibility of producing a particular drug, right, by this bioinformatics based approach, that is through homology modeling, wherein basically the variants whatsoever which are coming into picture with regard to the COVID, right? So the spike protein means the variations are taking place in the spike protein. So there is any possibility of producing a drug which can camouflage, means which can act as a camouflage like thing?
Yes, excellent question. A lot of studies, a lot of research is ongoing. Yes, there is a possibility. The problem is how effectively that's going to work in the human body is not quite clear. Let's say you're asking, your thought process is very smart. The thing is, you're trying to, let's say this receptor binding motif, right? You know, the mutations are happening in the receptor binding motif, and then can we take that part and then utilize that as a drug molecule to protect, right?
Yes.
Based on the homology. So that's a protein. When you give the protein, when you deliver a protein inside the body, so there is a lot of enzymes called proteases in the human body. They recognize any unknown proteins, they chew them, they just delete them from the body, remove from them the body, and before even start acting against the virus or against anything else. So that is a major problem that research experiencing.
Okay.
Right, so that's why there's a lot of it's not only making the drug. Computationally, you can make the drug. So a lot of questions I get about why computationally developed drugs are not effectively working. Yes, computational development is quite different. It's not a real-time environment. Real-time environment is quite different from the computational chemistry, right?
Yes.
So yeah, like I said, delivery method is also very important. That's why, spike protein is very well, again, spike protein synthesized in the lab, but it is protectively delivered using the lipid nanoparticles, right? So let's say 100 particles are delivered, maybe 90% of the particles are working and 90% of the protein is properly delivered and 10% is removed by the body probably. Who knows what's happening inside the body. So that's why we do these clinical trials and efficiency studies. First, we do the toxicity studies in animals before we go into the humans.
Yes. Sir, my another question is, my third question, if at all I'm not taking your time, sir. The third
No,
question is
I'm here for take all the participants. I would like to say take as much as time you want. Today, I'm happy to stay here another 1-2 hours and then answer all of your questions before I leave, unless Satavahana University has some objection.
Okay, sir. Sir, with my third question is with regard to the nasal vaccines, which are also in this vaccine production pipeline. Sir, the level of, just I want to know what is the level of this particular immune protection it offers when we compare it with the other particular vaccines which are available and also run in the market, and what is the level and span of the the immune response of these particular vaccines, the immune protection and the immune response.
Good question. So the nasal vaccines, right? The best advantage for the nasal vaccine is you don't have to inject into the body, so you don't feel the pain, number one. And for children, for kids, it's easy to give, number one. That's the best advantage for that. The immune response, it's very good for the respiratory viruses. So respiratory viruses, the main part that infects the human body, the respiratory viruses goes inside the body is nose, mouth, right? So if you generate the strongest immune response, robust immune response inside that part, the nose and the throat, before the lung, this is called upper respiratory tract. If you develop and get the force ready in the upper respiratory tract, it will not go down to the lower respiratory tract, which is the lungs. And it will not infect the lungs, and then you may not have to go towards the serious disease. It will not go towards the serious infection and disease and death. So nasal vaccines may produce even robust and better immune response compared to the because you're giving the injection on the hand, and then there are some cells, and then it has to travel through the body to go towards the lung or nose and airways. That's going to take a little bit of time compared to the nasal vaccine. So for the respiratory viruses, it all depends on the virus type. For respiratory viruses, which is flu virus, the coronavirus, all these type of viruses, nasal vaccines are more effective, but it's not as straightforward as the other vaccine development. That's why it takes some time.
Okay, sir. Thank you so much, sir. Thank you so much for answering my queries.
My pleasure. GK Ramana garu,
sir, good morning, sir. Good morning, sir. Sir,
good morning.
After COVID, ఎన్ని రోజుల వరకు యాంటీబాడీస్ డెవలప్ అవుతాయి? నెంబర్ 1, ఎన్ని రోజుల వరకు ఉంటాయి యాంటీబాడీస్? ఇంకోటి, వ్యాక్సిన్ తీసుకున్న తర్వాత యాంటీబాడీస్ ఏ ఎగ్జామ్ చేయించుకోవాలి టెస్ట్? ఇది కన్ఫ్యూజ్ ఏంటంటే యాంటీబాడీస్ దీనిలో ఉంటున్నాయి, దానిలో ఉంటుంది. యాంటీబాడీస్ స్పైక్ ప్రోటీన్స్ లో వస్తున్నాయి, యాంటీబాడీస్ దీనిలో వస్తున్నాయి. కాబట్టి దేనిలో ఏది కరెక్ట్? ఎన్ని రోజుల తర్వాత టెస్ట్ చేయించుకోవాలి?
సో ఫస్ట్ ఆన్సర్ మీ క్వశ్చన్ ఏంటంటే, మనం వ్యాక్సిన్ తీసుకున్నాక ఎన్ని రోజుల దాకా యాంటీబాడీలు ఉంటాయనే దానికి నేను సమాధానం చెప్తాను. ముందు ఇప్పుడు మనకు వచ్చిన స్టడీస్, ఎంఆర్ఎన్ఏ వ్యాక్సిన్లు కానివ్వండి, కోవిషీల్డ్ కానివ్వండి, కానీ మనకేంటంటే 1 సంవత్సరం నుంచే ఇస్తున్నారు కదా ఈ వ్యాక్సిన్. మనకు సమయం పోయిన కొద్దీ తెలుస్తుంది ఎన్ని రోజులు ఉంటాయన్నది. ఇప్పుడు ఈ క్లినికల్ ట్రయల్స్ లో పార్టిసిపేట్ చేసిన వాళ్ళందరినీ వాళ్ళు ట్రయల్ స్టార్ట్ అయిన రోజుల దగ్గర నుంచి 2 సంవత్సరాల వరకు వాళ్ళని పరీక్షిస్తూనే ఉంటారు. ఎవ్రీ నెలకోసారి ఎట్లా ఉన్నాయి అని చెప్పి పరీక్షిస్తూనే ఉంటారు 2 సంవత్సరాల వరకు. దాదాపు ఇప్పటి వరకు మనకు వచ్చిన డేటా అంతా 1 సంవత్సరం వరకు మాత్రమే ఉంది కాబట్టి 8 నెలలు, 6 నెలలు కానీ 8 నెలల నుంచి కానీ 12 నెలల వరకు ఉండే అవకాశాలు ఉన్నాయి. ఎక్కువ కొంత డేటా ఉంది, సమాచారం మనకు సమయం పోయిన కొద్దీ తెలుస్తుంది. ఒకటి. ఇక రెండవది వచ్చేసి, మీ రెండవ ప్రశ్న ఏంటంటే అసలు యాంటీబాడీలు వచ్చినా లేదా టెస్టులు, ఏ టెస్టులు చేయించుకోవాలని అడుగుతున్నారు. సో ఈ వ్యాక్సిన్ డెవలప్మెంట్ ఈ క్లినికల్ ట్రయల్స్ లో భాగంగా ఏం చేస్తారంటే 2-3 రకాల పరీక్షలు చేస్తారండి. యాంటీబాడీలు డెవలప్ అయితే మనం ఈ వ్యాక్సిన్ తీసుకుంటే ఫస్ట్ 2 రకాల రెస్పాన్స్ ఇస్తుంది ఇమ్యూన్ సిస్టమ్. ఒకటేమో హ్యూమోరల్ రెస్పాన్స్ అంటారు, అది యాంటీబాడీలని ప్రొడ్యూస్ చేస్తారు. నేను ఇందాక చెప్పినట్లే రకరకాల యాంటీబాడీస్, ఒక్క రకమే కాదు, వందల రకాలు, అసలు స్పైక్ ప్రోటీన్ వైరస్ ని చుట్టుముట్టే రకాల యాంటీబాడీస్ అన్నింటినీ తయారు చేస్తుంది. ఒకటి. రెండోది వచ్చేసి సెల్యులార్ ఇమ్యూనిటీ అంటారు. 2 రకాల సెల్స్ ని తయారు చేసి పెడుతుంది ఈ వ్యాక్సిన్. వాటిని మెమరీ టీ కణాలు, మెమరీ బి కణాలు అని అంటారు. ఆ మెమరీ టీ కణాలు, ఈ మెమరీ బి కణాలు ఏం చేస్తాయంటే అవి రిజర్వ్ ఆర్మీ లెక్క ఇవి వైరస్ యొక్క స్వరూపాన్ని, వైరస్ యొక్క స్వభావాన్ని గుర్తు పెట్టుకుంటాయి.
ఈ కరోనా వైరసా, జీకా వైరసా, లేక డెంగ్యూ వైరసా, ఏ వ్యాక్సిన్ ఇస్తే ఆ వ్యాక్సిన్ సంబంధించిన వైరస్ ని గుర్తు పెట్టుకొని నెలలు సంవత్సరాల తరబడి బాడీలో రిజర్వ్ ఆర్మ్ లెక్క నిల్వలో ఉంటాయి అన్నమాట. ఎప్పుడైతే ఆ మళ్ళీ వైరస్ ఇన్ఫెక్షన్ బాడీ లోపలికి వస్తుందో వెంటనే ఈ రిజర్వ్ ఆర్మ్ లెక్క ఉన్నటువంటి ఈ మెమరీ టీ కణాలు, మెమరీ బి కణాలు యాక్టివేట్ అయిపోయి వెంటనే రకరకాల బి కణాలు ఏం చేస్తాయంటే బి కణాల పని యాంటీబాడీలను జనరేట్ చేసి వైరస్ మీద అటాక్ చెప్పేయడం. ఈ టీ కణాల పని ఏంటంటే అటాక్ చేయబడ్డ వైరస్ ని తిని బయటపడేయటం, లేదా వైరస్ అటాక్ అయిన సెల్స్ ని చంపేసి బయట తీసేయడం, ఎందుకంటే వైరస్ కి ఇన్ఫెక్షన్ పెరగకుండా చేయడం. సో ఆ రిజర్వ్ అనేది డెవలప్ చేసి పెడుతుంది ఈ వ్యాక్సిన్. సో మనం ఈ కరోనా వైరస్ దానికి వచ్చేసి 2 డోసులు ఎందుకు తీసుకో, 2 డోసులు అందుకోసం తీసుకుంటాం. ఎందుకంటే మొదటి డోసు ఇచ్చినప్పుడు ఒక రకమైన మెమరీ కణాలు, మెమరీ టీ కణాలు, బి కణాలు తయారు చేయబడతాయి కదా. అయితే వాటిని తక్కువ కాలం ఉంటాయని చెప్పేసి 2వ డోసు 4 వారాల తర్వాత 2వ డోసు ఇస్తే ఈ 2వ డోసు ఏం చేస్తుంది, ఆ ముందు డోసు తయారు చేసిన మెమరీ కణాలని, మెమరీ టీ కణాలని, మెమరీ బి కణాలని యాక్టివేట్ చేసేసి ఇంకా ఎక్కువ కాలం నిల్వ ఉండే యాంటీబాడీని, ఇంకా ఎక్కువ కాలం నిల్వ ఉండేటువంటి మెమరీ టీ కణాలు, మళ్ళీ కొత్త మెమరీ టీ కణాలని, కొత్త మెమరీ బి కణాలు తయారు చేసి బాడీలో సంవత్సరాలు నిల్వ ఉండి. అందుకని 2 డోసులు తీసుకోవడం చాలా ఇంపార్టెంట్.
ఇంకోటి ఎక్స్ట్రా తీసుకోమంటారు,
రెండోది వచ్చేసి
థర్డ్ డోస్ తీసుకోమంటారు.
లేదండి, ఇప్పటి వరకు లేదు. థర్డ్ డోస్ ఇప్పటికైతే అవసరం లేదు, ఈ 2 డోసులే సరిపోతాయి. అయితే ఇంకొకటి ఏంటంటే ఇప్పుడు ఈ క్లినికల్ ట్రయల్స్ లో భాగంగా యాంటీబాడీస్ ఉన్నాయా లేదని టెస్ట్ చేయడమే కాకుండా యాంటీబాడీస్ సమర్థవంతంగా వైరస్ ని ఆపుతున్నాయా లేదా అని చెప్పేసి సూడో వైరస్ అంటే కరోనా వైరస్ లాంటి వైరస్ ని ల్యాబ్ లో తయారు చేసి దాని మీద టెస్ట్ చేస్తారు ఒకటి. రెండోది వచ్చేసి నిజమైన కరోనా వైరస్ ని ల్యాబ్ లో తయారు చేసి దాని మీద టెస్ట్ చేస్తారు ఒకటి. ఇంకా మూడోది వచ్చేసి నిజంగానే ఈ మెమరీ టీ కణాలు, మెమరీ బి కణాలు ఉత్పత్తి అవుతున్నాయా లేదనేది ఈ టెస్టులు చేస్తారు. ఈ టెస్టులు అన్ని కూడా చాలా కాస్ట్లీ టెస్టులు, ఇవి బయట ప్రపంచంలో ఉండవు. ఓన్లీ ఆ యాంటీబాడీలు ఉన్నాయా లేదా టెస్ట్ చేసే పరీక్ష మాత్రమే బయట ప్రపంచంలో ఉంటది. కాబట్టి అసలు ఆ టెస్ట్ నా దృష్టిలో, నేను ఈ మధ్య ఒక తెలుగులో ఒక వీడియో తయారు చేసి పెట్టినాను, మీరు బహుశా మీకు నేను పంపిస్తాను, మీకు ఉంటే అది ఎట్లా అంటే కాంటాక్ట్ చేయండి ఆఫ్లైన్ లో, నేను పంపిస్తాను మీకు. అయితే దీని వలన మీరు పోయి అనాలిసిస్ అది యాంటీబాడీ టెస్ట్ ఏదైనా ల్యాబ్ లో చేయించుకుంటే ఒక లెట్స్ మీకు చాలా మస్తు ఒక ఫుల్ వచ్చినాయి అనుకుందాం, మరి అన్ని కలిపి వైరస్ మీద అటాక్ చేస్తున్నాయా తెలియదు, మనకు తెలియదు. ఏం రాలేవు అనుకుందాం, టెస్ట్లు ఏం రాలేవు అనుకుందాం, మరి అవి ఆల్రెడీ జనరేట్ అయి ఉంటాయి. ఇప్పుడు ఏంటంటే బయట టెస్ట్ చేసే అని ఒక ఎలీసా టెస్ట్ అంటారు, దానికి 60% మాత్రమే ప్రామాణికత ఉంటుంది. ఇది ఎట్లా అంటే చేపలు, ఇప్పుడు చెరువులో చేపలు పట్టినట్టు పోయి వల వేస్తే దొరకని ఆ టెస్టులు అట్లాంటివి అనమాట. క్లినికల్ ట్రయల్స్ లో జరిగే ప్రయోగాల ఫలితాలు ఈ టెస్టులు చాలా రోబస్ట్ గా ఉంటాయి. సో అందుకని సైన్స్ నమ్మండి, మెడిసిన్ నమ్మండి, మీరు ధైర్యంగా ఉండండి అంతే. అదే కానీ ఇవి అనవసరమైన టెస్టులు, ఇవన్నీ పైసలు వేస్తూ లేనిపోని తలనొప్పి అండి. ఇట్లా యాంటీబాడీలు వచ్చినాయా లేదని టెస్ట్ చేయించుకోవడం మీరు. కాకపోతే ఏంటంటే అందుకే చెప్తున్నాం, ఇప్పుడు ఈ వేరియంట్లు అన్ని కొత్తగా వస్తున్నాయి కాబట్టి మాస్క్ పెట్టుకోవటాలు, సోషల్ డిస్టెన్స్ పాటించడం, ఇవి చేయడం అనేది చాలా ముఖ్యంగా ఏజ్ ఎక్కువగా ఉన్న వాళ్ళందరికీ కూడా అది చెప్తున్నారు.
సరే, ఇంకోటి సిటీ వాల్యూ కూడా వ్యాక్సిన్ తీసుకున్న సెకండ్ డోస్ అయిపోయిన తర్వాత కూడా 40 days తర్వాత కూడా సిటీ వాల్యూ 12-13 కూడా వస్తుంది సార్. అది ఎట్లా అన్నట్టు, ప్లస్ సివియారిటీ కూడా ఎక్కువనే ఉంటుంది పేషెంట్స్ కి.
వ్యాక్సిన్ తీసుకున్న
12 ఇంజెక్షన్స్ కూడా ఇస్తున్నారు.
అంటే మీరు వ్యాక్సిన్ తీసుకున్న తర్వాత ఇన్ఫెక్షన్ వచ్చిన వాళ్ళకి అంటున్నారా?
అవును సార్, అవును. సిటీ వాల్యూ కూడా 12 to 15 వరకు వస్తున్నది వాళ్ళకి కూడా.
ఇప్పుడు మీరు సిటీ వాల్యూ దాని గురించి అంత ఎక్కువ ఇదవద్దండి. ఏమవుతుందంటే ఈ వైరస్ ఇన్ఫెక్షన్ బాడీలో పెరగడం అనేది ఒక్కొక్క వ్యక్తికి ఒక్కొక్క రకంగా ఉంటుంది. ఒక వ్యక్తిలో ఇప్పుడు ఇమ్యూన్ సిస్టమ్ ఏమైపోతుంది అని అంటే, అది యంగ్ గా ఉన్న వాళ్ళలో తొందరగా యాక్టివేట్ అయిపోయి రోజులలో అది వైరస్ ని తగ్గించేస్తుంది. అప్పుడు సిటీ వాల్యూ తక్కువైపోతుంది. కానీ వయసు పెరిగిన కొద్దీ వాటి వలన వచ్చే లక్షణాలతోని కొంచెం దీని యొక్క రెస్పాన్స్ తక్కువగా ఉంటుంది కాబట్టి వాళ్ళకి కొంత టైం తీసుకుంటుంది. ఇప్పుడు ఇది వీళ్ళకి ఎవరికి ఎంత అవుతుంది అనే విషయాలు తెలియకనే కొందరు సీరియస్ అవుతున్నారు, కొందరికి చాలా రోజుల తర్వాత సీరియస్ అవుతున్నారు. అదొకటి. ఇంకోటి ఏంటంటే యంగ్ గా ఉన్న వాళ్ళు కూడా బాగుంటుంది అంటే మళ్ళీ ఇప్పుడు ఉన్న వాళ్ళ ఆహారపు అలవాట్లు అనేవి చాలా ఇన్ఫ్లుయెన్స్ చేస్తాయండి దీన్ని. వాళ్ళ బాడీ ఉన్నటువంటి కొమార్బిడిటీస్ అంటారు వాటిని, అంటే షుగర్, డయాబెటీస్, హార్ట్ ప్రాబ్లం, వాళ్ళు ఇంకా చాలా రోగనిరోధక వ్యవస్థ యొక్క తగ్గించే మందులు వాడుతున్న వాళ్ళు ఉంటారు కొందరు, ఈ డయాలసిస్ పేషెంట్లు కానీ అట్లాంటి వాళ్ళు. సో వాళ్ళకి చాలా తక్కువ ఉంటుంది. అట్లాంటి వాళ్ళకి మీకు వైరస్ పెరిగిపోతూ ఉంటుంది. అప్పుడు సిటీ వాల్యూ తక్కువ వస్తుంది. వైరస్ బాడీ లోపల ఎక్కువైతున్న కొద్దీ సిటీ వాల్యూ తక్కువ అవుతుందండి. అది ఎట్లా అంటే అది ఒక వ్యక్తిని బట్టి ఉంటుంది, వ్యక్తి యొక్క శరీరాన్ని బట్టి ఉంటుంది. జాగ్రత్తలు తీసుకున్న వ్యక్తికి ఏమీ జరగదండి.
ఇంకా రెమ్డెసివిర్ ఇంజెక్షన్స్ రెగ్యులర్ గా ఇస్తున్నారు కదా సార్, ఇంత వ్యాక్సిన్ ఇచ్చినా కూడా 2 వ్యాక్సిన్స్ అయిపోయినా కూడా రెమ్డెసివిర్ 5 డోసెస్ ఇస్తున్నారు. అడ్మిషన్స్ అవుతున్నాయి, సివియారిటీ పెరుగుతున్నది. చాలా మందిని చూస్తున్నారు.
ఇదంతా వేరియంట్స్,
డయాగ్నోస్టిక్ సెంటర్.
ఎస్ అవునవును, అది వేరియంట్ వల్లనండి. వేరియంట్స్, కొత్త వేరియంట్స్ వస్తున్నాయి కదా. రెమ్డెసివిర్ అనేది అది చూడు, సీరియస్ కాకముందు రెమ్డెసివిర్ ఇస్తే ఉపయోగం ఉంటది, కానీ సీరియస్ అయిపోయిన తర్వాత రెమ్డెసివిర్ ఇచ్చిన ఉపయోగం లేదండి. సిటీ వాల్యూ మీరు చెప్పినట్లు అంత తక్కువ వచ్చినప్పుడు రెమ్డెసివిర్ ఇచ్చిన పెద్దగా ఉపయోగం ఏమి ఉండదు. ఇన్ఫెక్షన్ వచ్చిందని తెలిసిన తర్వాత ఇమీడియట్ గా ఇప్పుడు ఏంటంటే మనకి ఇప్పుడు వ్యక్తులకి ఈ రోజు వచ్చిన ఇన్ఫెక్షన్ నిన్న వచ్చిందా, వారం రోజుల కింద వచ్చిందా ఎవరికి తెలుసండి. కొందరికి అసిమ్టమాటిక్ ఇన్ఫెక్షన్స్ వస్తున్నాయి. సో వాళ్ళు మీ డయాగ్నోస్టిక్ సెంటర్ కి వచ్చిన రోజే అది ఆ రోజే కాకపోవచ్చు వైరస్ వాళ్ళకి ఇన్ఫెక్ట్ అయ్యింది. 3 రోజుల ముందు అయిందంటే సిటీ వాల్యూ హై ఉంటుంది, ఆ లోపల బాగా పెరిగిపోతుంది కదా.
ఓకే సార్, లాస్ట్ క్వశ్చన్ సార్. ఇది కోవిడ్ వచ్చిన వాళ్ళు రెగ్యులర్ గా మళ్ళీ టీబీ టెస్ట్ చేయించుకోవాలంటుండ్రు, అది అవసరమా సార్? దానికి దీనికి ఏమైనా సంబంధం రావడానికి?
టీబీ టెస్ట్, ట్యూబర్క్యులోసిస్ లేదండి, దానికి దీనికి ఏం సంబంధం లేదు. అది వేరు, ఇది
ఆర్టికల్ చదివాను, అది వస్తుంది.
దేంట్లో అండి, దేంట్లో చదివిన ఆర్టికల్?
నిన్న ఆర్టికల్ ఒకటి చూసినాను సార్, అందుకని అడుగుతున్నా.
న్యూస్ పేపర్లో అయితే కాదండి,
గవర్నమెంట్
లేదండి.
సెంట్రల్ గవర్నమెంట్
నాకు తెలిసిన సమాచారం, అవునా, నాకు తెలిసినంత వరకు అది ఏం లేదండి, అట్లాంటి అవసరం లేదు. ఊపిరితిత్తులు కొందరు ఆల్రెడీ టీబీ ఎఫెక్ట్ అయి ఉన్నోళ్ళు ఏదన్నా వాళ్ళు చేసుకోవచ్చు, కానీ లేని వాళ్ళు అయితే ఏం అవసరం లేదు. టీబీకి కోవిడ్ కి సంబంధమే లేదు. టీబీ వచ్చేది బ్యాక్టీరియాతో, ఇది వైరస్ ఇన్ఫెక్షన్, అది వేరు ఇది వేరు. అండ్ చాలా హ్యాండ్స్, లాట్ ఆఫ్ హ్యాండ్స్ ఐ కెన్ సీ దట్. నెక్స్ట్ ప్రొఫెసర్ అశోక్ గారు, సార్ unmute yourself and you can ask question.
Sangeeta, hello.
Yeah,
Sangeeta gar. Yes, Sangeeta gar.
Sir, a small doubt I'm having. Previously also we had many viral infections, no sir, like dengue or swine flu and AIDS viruses and etc. So many other viruses, but they are not undergoing any mutation or any new variants are being found, though in spite of vaccinations was not available to them. But why this COVID virus alone is undergoing so many mutations and new variants are generated, sir?
Good question. Very good question. So the thing is, RNA-based virus. Coronavirus is RNA-based virus, number 1. Number 2, it's a respiratory virus, like similar to the flu virus, common cold virus type of thing. So the tendency of RNA viruses is high mutation rate. So RNA viruses, they undergo heavily mutations. The only good thing for the coronavirus is the virus genome is very big. Even if the flu vaccines, the flu vaccines, every year, every couple of years, we have to make new vaccines, because flu viruses automatically undergo mutations regularly, respiratory viruses, because they transmit to the people very easily through the respiratory tracts, right? By, nose, now aerosols, nose and mouth. This respiratory virus infection rate is very high. That's why they go under mutations very heavily. But HIV transmission is HIV virus also undergoes mutations. So that is also an RNA virus. But the mutation rate is slow because the infection rate is also slow, number 1. And the dengue and other viruses, some DNA viruses are that they don't go under mutations as rapid as the RNA viruses. So it all depends on the virus type. I hope I answered your question.
Yes, sir. Sir, one more question, sir. The person who is already infected with COVID, do they need to take again shots of vaccine, sir?
Yes, they do have to take the vaccine. Yes.
Okay, sir. Thank you, sir.
Yeah, because see, you once you get the infection, you develop certain amount of the immunity. But if you want to make it more robust, you definitely need to take the vaccine to develop the memory T cells and memory B cells that will stay as a reserved army to fight against new infections for longer periods of time.
Okay, sir.
Sir, there is one more question, sir. Efficiency of Novavax.
Go ahead.
With regard to the biocompatibility and toxicity issues.
I think I just answered that question for Preeta Sharma. Somebody asked. It's efficient vaccine. It's working perfectly fine. In phase 3 clinical trials, it showed 89% efficiency. The biocompatibility, I mean, there's no issues with the biocompatibility because preclinical studies, it's proved safe and non-toxic. And that's why it's moved to the phase 1, 2, 3 clinical trials, right? So they're using the lipid nanoparticles, which are widely used to deliver the vaccines or other type of medications. So there's no issues with the biocompatibility.
There is one more question, sir. Efficacy of a nasal vaccine compared to the other vaccines towards the immunity response.
I think I also answered this
Sir,
same
you
question.
answered, sir. It was the same question.
Yes, I answered all these questions. Yeah, I answered all the questions. Let's move on to the next person. Kiran Kumar. Kiran Kumar may ask the question. He raised the hand. Go ahead. Unmute yourself and ask the question.
Yes, sir. Good morning, sir. This is Kiran. Research scholar of Mechanical Department.
Say it again.
Research scholar of Mechanical Department of Usman University. Yeah,
Which university?
Yeah, Usman University.
Okay.
Yeah, sir. I have a simple question, small question, sir.
Go ahead. Go ahead.
Yes, sir. There are many rumors in the society about the side effects, long-term side effects. Sir. Are there any long-term side effects after the vaccination? Sir?
Very good question. Let me answer that. See,
yes.
So longer-term side effects, we're still studying, right? Like I just answered the question. You know, when you have a side effect, during the phase 3 clinical trials, the participants will be monitored for 2 years, next 2 years. But however, regardless of that, any side effect, longer or shorter, any side effect, if you don't experience within 30 to 45 days, you would, I don't think you will experience. See, for example,
sir.
Have you ever heard that, somebody joined in the hospital after 2 years of taking a vaccine saying that I had a side effect because of the vaccine I took 2 years ago?
No.
These instances are not possible, right? So that's why, FDA and DCGI, they put this clear rule saying that phase 3 clinical trials, after 2 months of the 2 doses, the data we need to see after 2 months of the 2 doses. So they added 2 more extra weeks rather than 45 days. They added 2 more weeks after 60 days of second dose administration. We need a safety data, efficiency data, immunogenicity data, and based on that, these vaccines are authorized. So that's why what I'm recommending to the people is, even though each individual is different, of course, we have done is 30,000-40,000 people phase 3 clinical trials. Sometimes, most importantly, people with the comorbidities like diabetes, dialysis, hypertension, kidney issues, heart patients. So what I would recommend them to do is, you have to monitor your own health for 2 months after taking the second dose of vaccination to see if something is happening. Let's say if something unusual is going on, just go and see the doctor immediately. Otherwise, you're fine and safe.
Okay. Yeah.
Sir Mahendra Ji.
We'll go to the next.
Good morning.
Go. Yeah,
I am
morning.
Dr. Viraya
from
right? Dr. Viraya, Department of Chemistry, some affiliated
yes,
college, SAP College, Vikharabad, just about 70 kilometers away from Hyderabad. So,
yes, sir.
Mahendra Ji, many doubts are cleared by your presentation regarding this variants, virus, and waves. I have one doubt regarding the gap between first and second dose of vaccination. First and the gap period between first and second dose of vaccination.
Yes.
So does it depend, that gap time between these 2 doses depend upon the government policies or efficacy of the vaccine or availability of vaccine? I have this doubt. Just
yeah, sure. I can answer that question. So yeah, that's a very good question. Actually, right now there is a lot of confusion going on based on the vaccines that we have in India, right? So it depends. There are various different factors. The most important factor depends on the scientific data. It's not the government policy or government or anything else. First most important thing depends on the scientific data. Second thing is vaccine availability. Absolutely right. In UK, what happened was in the beginning there was not enough vaccine available, so the government decided to give at least one dose for every most of the people so that they can be protected. That's one of the things. I mean, see, even single dose also provides certain amount of protection. It may not be the robust protection, but certain amount of protection certainly protects, gives the efficient protection.
Okay.
Right. So for the Covishield vaccine, later on they have done all these studies, because Covishield vaccine is one of the first vaccines who authorized in the beginning days, right? So we have a lot of data available. So the gap you may take from 4 to 12 weeks. Anytime you can get that vaccine, it's fine. But again, the government policy, if they have a more vaccine available, they can cut it down to 8 weeks or 6 weeks or 4 weeks.
That's
So
what actually happened in
At
India.
least 4 weeks. Yeah, at least 4 weeks is required. But at most, as of now, 12 weeks is the data for the Covishield. 4 to 6 weeks is for the Covaxin. Sputnik, it's not, still for 4 weeks only. There's no data available. The other vaccine is mRNA vaccine, still 4 weeks. So only Covishield is the one that the gap is being scientifically proved from 4 to 12 weeks.
Okay, sir. Thank you, sir.
Yeah, my pleasure. And next, Mr. Raju, yes, please unmute yourself and ask the question.
Hello. Yeah, good morning.
Yes, go ahead.
Good morning, sir. This is Raju from Osmania University, sir. So I have a small doubt regarding what is the effect of third wave on children, sir. So if it affects, what are the drugs available for the children, sir?
The children's, see, I mean, how this virus or wave is going to behave, it's hard to predict, number one. But based on the previous data, let's say the first wave, the virus impacted in the people above 60, 55, 60 years old, right? The first wave. But middle age and younger children were not at all impacted in the first wave. In all over the world, same thing. That was a surprise. And when the second wave came in many different countries, including the middle-aged people who are also infected, but not the children, right? So the virus, why middle-aged people are infected? Because old-age people started getting vaccination. So they have started getting some immunity from the vaccines. So they're protected. Even though they're infected, they're not dying. But middle-aged people who are not vaccinated, even though they're infected, they started dying in there. So in the second wave, right? Third wave, there's a possibility, but I don't see that possibility because the children have the strongest immune response in generally. The children in the sense above a certain age, not the infants or babies, right? But still, again, we should not take a chance. We have to protect our children. Means we have to follow the COVID appropriate behavior, even with the children. So that's the best thing to do, rather than, and also medications. Right now, the only one medication against the COVID-19 is remdesivir. And the second thing is monoclonal antibody therapies, which is very expensive. These two are right now available there as a therapeutic options. And other than those two, the vaccination. And vaccines for children is not yet available in India. It may take few more months. Until then, we have to protect the children. Any wave, you never expect how virus is going to behave in the next wave. So being safe is better than not following the proper attitude, proper behavior, COVID appropriate behavior.
Thank you, sir. Thank you very much.
Professor Ashok, sir. Ashok, sir, do you have any questions? Professor Ashok, sir.
Sir, good morning, sir.
I see you are. Yeah, good morning.
Sir, good morning, sir.
Can you introduce yourself? Yeah, go ahead. Good morning. Introduce yourself.
Sir,
please.
I am Dr. B. Sridevi from YA Government College. Sir, I have one doubt.
Hello, good morning.
If a person attack and recover from the first coronavirus, first COVID, after that, unfortunately, he attacked again present Delta virus. Suppose he get vaccinated, how much extent of his efficacy of his Covishield vaccine it works?
Okay. Yeah. So there's a chance of already infected people, usually they develop antibodies and memory cells to fight with the new infections. It all depends on that person's body. If he has some other comorbidities, or if he has a weaker immune system, or if he has some other organ transplantation has been done, if he's using the immune suppressing medications, then there's a chance of getting a second infection as well. But vaccine also helps them to protect for the from the second infection. So it makes the immune system stronger. All vaccine does is it improves the immune system's strength. It makes it more and more stronger. But also that process is also depends on each individual. So people, let's say a healthy person, it's good for the healthy person. But a sick person with different comorbidities, the efficiency may be a little bit lower, but still it protects strongly.
Excuse me, sir. This is situation. What in case of that C-section that ladies, some people they are got major operations. In case of ladies, what about that efficiency of this COVID vaccine? This.
So you're asking the ladies who already had the C-section and babies, right? Or in the past,
Yes,
C-section you're asking. Or new mothers, or in the past C-section? Your question for new mothers? In the past is not a problem. There's nothing happens to them. It's highly efficacious, and it's as efficacious as the normal.
Okay, thank you, sir.
Okay.
Sir, one more question. Sir?
Yeah, please go ahead. Go ahead.
Sir, now whatever vaccinations we are administering to the common people, suppose after 2 or 3 years, again if a new variant in COVID arises, then do these vaccines or antibodies naturally which have developed, will they be offering protection against this new variant, sir?
So they should in general, but as not efficient as they are now. They may not, because that's why we develop new vaccines pretty much every other year for the flu virus, because the flu virus mutates very fast and its genome is very small, and the mutations are rapidly changes its the virus's structure. But for the coronavirus, I don't think that would happen, because coronavirus genome is very large. It's 30 kilodaltons. It takes long period of time, and again, these variants, these small mutations are common in viruses. The mutations, the virus's nature is mutating. So it's a survival stress. So virus wants to survive from our induced stress from vaccines and medicines, right? Only method it has is mutating itself, becoming new variants. So we can protect that. We can resist that by doing two important things. Number one is vaccination, rapid vaccination. Number two is COVID appropriate behavior, following the non-pharmaceutical interventions. And virus can also always mutate, not only strong mutations. Virus, when virus mutates, it can mutate in a stronger way, or it can mutate towards a weaker way. When it mutates to stronger way, it becomes more and more stronger, and it becomes more new variants. When it mutates towards a weaker way, virus becomes weak and weak. It will stop its infectivity, and it will go away from the human population. It will go and find a different host and silent itself. So the two things will process. Two processes are possible in the virus mutations. So for next 2-3 years, I wouldn't worry about next 2-3 years. Right now, we need to get over with this pandemic by vaccination and following the COVID adaptive behavior.
Okay,
sir. I hope I answered your question.
Yes, sir. Thank you, sir. Sir, one more question. Right now, whatever protocol the MD physicians are following for treating COVID patients at home isolations or something like that without hospitalization, for them, they are being administered this azithromycin and doxycycline, some antibiotics. So how far these antibiotics can kill virus, sir?
No,
is it efficient
antibiotics.
for that?
Yeah, the problem is, this is again, administration problem. The antibiotics doesn't effective at all against the virus. In the US, FDA clearly mentioned this. Ivermectin is not working. Azithromycin, doxycycline, they're all useless. I mean, in the beginning, like last year when the pandemic started, no one knew what's happening in the human body because of this virus infection. So they started giving all different types of antibiotics, antiviral agents, whatever is available, approved, to see that's going to help the patients. But now we have better medications, better therapies like monoclonal antibody therapies, antibody cocktail therapies we have, and remdesivir. I mean, the problem is remdesivir is an injection. You have to go to hospital to get that. Otherwise, if we have oral, if it is an oral pill, it could have saved a lot of people. And again, one more new oral pill is coming, which is called molnupiravir. It's probably in September, October, we'll get the final data. Hopefully by end of this year, that is available to treat these infections. And that's an oral pill. It's you can take it by mouth, and you don't have to go to hospital. If that is approved, then again, when you find that, when you find you got infected or you are detected with a positive infection, when you start if you start this medication, remdesivir or molnupiravir, it will not increase the virus population. So the way these medications work is they stop the replication of the virus. The mechanism that they work is that they'll stop the replication. Replication means increasing the virus number. They stop it. When you stop the replication of the virus, it will not increase the disease severity. So that's when you control the virus replication, and the immune system will act against the virus, and then it will clear some of the virus from the body. That's how it works. But unfortunately, for the second wave time, hospitals are flooded with the serious infections. And then the other problem is, this virus infection, when it's going to go towards the serious, it depends on each individual. Some people 2 days, some people 5 days, some people 3 days. It's hard to be kind of predict. So that's why lots of people were got the serious infection. But still, these are the medications only. But antibiotics, all that stuff is even some other medications, they're useless.
Okay, sir. Thank you, sir.
Dr. Mahendraji. Dr. Mahendraji,
go ahead.
Now everybody is talking about vaccines, but ultimately we require a new drug for coronavirus. What is the status of drug discovery presently going on?
Sure.
Yes.
Sure. Yes, sir. There's a lot of research going on in the drug discovery side as well. Approximately more than 500 potential candidates are under development, the research. Okay. And among those more than 500, more than 400 of them are already in the clinical trials, phase 1 to 3 clinical trials.
Nice.
As I mentioned, molnupiravir, another new drug from Merck Pharmaceuticals together, Ridgeback Bio, together they developed from Emory University here in the US. It's an oral pill. It also works similar to the remdesivir.
It is known or
phase
unknown?
2.
It is known
it's
or
a
unknown?
known.
Known. Okay.
Known. It's a known, but it's not yet authorized. It's not yet approved.
Okay.
But the phase 3 clinical trials are ongoing. The results may come September. So August, September, October time. So we will get that approved probably. But it's only acts mildly infected patients. It's not for the severe patients, because, sir, excuse me, this virus is attacking 2 ways. Number one is it's utilizing our own immune system against our body.
Okay.
So when immune system activated to attack the virus escapes from that, and then this immune system go and attack the lung cells, and then started killing the lung cells. Then lung cell damage is causing the lung problem. So that's why people are using steroids to recover from that lung issue. So controlling the immune system. Steroids controlling the immune system to not to cause the damage to the lungs. So multi-drug therapy is required for the severely infected patients. But mildly infected patients, mild, moderate patients, this remdesivir type of medications are fine. But there's a lot of research ongoing, sir.
Okay. Thank you.
Dr. Hari Padma Sridhar wants to ask a question. Please go ahead, ma'am. Ask your question. Unmute.
Good morning, sir. I have one.
Yes. Good morning.
I don't know if it's a kind of a silly question, because I'm from a non-biology background. What I just want to ask is, See, the virus, it is attacking by, through its spike virus, and we have the medicines working on, people are working on, to dealing that spike and to protect the body from the virus and sort of thing. So is there a possibility that, you can work on something to destroy, for a kind of a self-destruction of the virus? If you can initiate kind of a self-destruction, the virus itself, how it's replicating, we're trying to restrict its replication. So if you can, trigger kind of self-destruction of the virus, is it possible, sir?
Yes, ma'am. Great question. Another great question. Exactly. That's what is remdesivir doing, the self-destruction. So it's stopping the virus to reproduce itself. So what it does is, it's a replicase inhibitor. So the remdesivir, so there is an enzyme. This virus has a unique enzyme which is called replicase or RNA-dependent RNA polymerase. That enzyme makes this positive sense RNA of this virus to a negative sense RNA. That means it's creating a template, prototype. And from this prototype, other enzymes from the body will start copying Xerox copies of that particular prototype, make more and more Xerox copies. If we can stop this prototyping, then the virus cannot make more and more prototypes, right? So the replicase, this enzyme is crucial enzyme, and the remdesivir stops that replication. It goes and binds to one of the binding site of the replicase enzyme, and it stops the replicase activity. That's how it works. And there are so many other also. I mean, I can. This additional answer to Professor Ashok's question is, recently Pfizer announced another molecule. It's a protease inhibitor. This acts on during the infection time. When the virus goes and attaches to the ACE2 receptor, after that there are some enzymes called proteases, cleaves the spike protein, making it easy to infect the cell. And if you inhibit those protein enzymes also, we can effectively control the virus infection. So there is a new molecule. It's PF70333312. So it's a new molecule. Phase 1 clinical trials are ongoing. It's totally new. And molnupiravir, again, I said, it's called EIDD2801. Emory University developed initially, not developed. This is a repurposed drug. So they are developing this against the Ebola virus, and they tested it towards corona, and then which showed a good activity. Remdesivir also same thing. Remdesivir was developing for Ebola virus. When they tested against coronavirus, it showed some activity, and then they started clinical trials with coronavirus patients. So molnupiravir, again, one more drug molecule hopefully will be coming soon, which is also a replicase inhibitor, which will stop the virus replication. That means self-destruction.
Thank you, sir. Thank you.
Welcome.
Any more questions?
Yeah, please go ahead and yeah, go ahead.
Any more questions?
There are some chat questions on the chat window. I see. Can somebody read the questions in the chat window? I'm happy to answer those too.
Sir, already these have been asked, sir. They are repetitive questions.
Oh, okay. Okay.
Thank you very much. Thank you very much for your clear explanation of the efficacy of different vaccines on different variants of virus. Clearly differentiated between the authorized and unauthorized vaccines available, and been demonstrated the scientific evidence on vaccine benefit and impact of virus on probability of hospitalization, and also very clearly shown the scientific reasons for vaccines limitation, and also the statistical evidence of impact of vaccines in reducing the death rate. Also, once again highlighted the COVID protocol. Thank you very much, sir, for the very enlightening message of this topic. Thank you very much, Mahinder B. Diwalji, for sparing the valuable time.
Dr. There's a one question I see on the chat window. I'll quickly answer that before we close.
Sir.
Somebody, it's Mr. Nitin Kumar, asking that can you please briefly explain about post-COVID vaccination? As per ICMR guidelines, a post-COVID patient should take his vaccination only after 3 months, whereas some of the local doctors are suggesting to take vaccine after 2 months. How does this affect a post-COVID patient immune system? Great question, actually. This is a very good question. So post-COVID, in the sense, a person who first infected with the COVID and then he was cured, then when he can get the vaccination. So anybody who ever got the infection, once they're, according to the CDC, Center for Disease Control Prevention guidelines, they can get the vaccine right after they're cured. Once they complete their isolation period, once they complete their quarantine period, once they feel that they're not infecting other people, then they can go and get the vaccination. But whoever got the plasma therapy, whoever taken the plasma therapy, whoever taken the monoclonal antibody therapy, they have to wait for 3 months, 90 days after they can get the vaccine. So that is the only, but ICMR guidelines, I'm not sure about, and local doctor guidelines, I'm not sure, but according to the Center for Disease Control and Prevention, anybody who infected with the COVID-19, once they recovered, they may get vaccine. But only people who treated with monoclonal antibodies and plasma therapy, they should wait until 3 months, 90 days.
Sir, they also need to take 2 shots, sir, of vaccines after they got recovered?
Yes, so 1 dose also is effectively working on them. If 2 shots are available, it's good to take. If the 2 shots are not available, then 1 shot is also good, sufficient for them. And another question I see is what precautions to be taken during the domestic flight travel?
Yes.
Kindly explain. Yeah, domestic flight travel, same thing, COVID appropriate behavior. No, it's not only the flight travel, any travel, bus travel, auto travel, taxi, any travel. You're going out of the house, even though you're vaccinated, you put mask on yourself. Masking is most important, and a sanitizer in your pocket. And try to keep social distancing. That's what is also very, very important. The other question I see is what is black fungus? Why is it affecting on COVID recovery patients? Please explain, sir. Sir, can I go ahead and answer these questions? Professor Ashok, sir.
Yes, you can.
Okay. See, the black fungus, yes, very good question, right? So the black fungus is a fungus infection. When any person who infects by a fungus or a bacteria, when their immune system is not effectively fighting against those fungus or those bacteria, those virus, right? So many patients after COVID, they are getting this black fungus because they are inappropriate use of the steroids. The steroids can be used by a medical practitioner. They can be prescribed. They can be given by a medical practitioner. You just cannot go and a medical store and buy and take the steroids. If you take like that, these kind of infections will happen. So, and also you have to maintain the hygiene. When you're taking steroids, you have to very, very clean. That is another important thing. Because if simple fungus, a simple bacteria can also attack your body. Because what steroids does is they reduce our immune system's capacity, because the coronavirus infections causing the rapid response of immune system, which is attacking our lungs. That's why lung damage is happening. So control that lung damage. Doctors are prescribing the steroids, but steroids are very highly controlled medications. They must be taken under the doctor's surveillance. You have to be communicating with the doctor, and you have to take as much doctor prescribed only. You cannot take yourself. That is most important to avoid this black fungus or any other kind of infections. I mean, see, for one, I'm surprised when I see this. The steroids are being administered against COVID infections from all over the world, everywhere in the world. But black fungus happened only in India. Think about it. Why? Number one could be inappropriate usage of the steroids, no hygiene maintenance, unauthorized usage of the steroids probably. So these are the reasons I can say. any medication, controlled medication, I would highly recommend to speak with the doctors. Especially, with the COVID pandemic, so many things are coming out. So much misinformation is communicating through the social media, YouTube, all those things. So be careful, trust the doctors and science and scientists. That's what I recommend. The other question is.
One more question, sir.
Go ahead, please go ahead. So
How
2DG.
2D.
Let me.
How
Yeah, I'm reading. I'm answering that question.
Yeah.
I'm answering that question. So Mr. Karimnagar is asking that how effective is 2DG drug development by DRDO? So how effective is not very well known, but it is not harmful. It is harmless. So I'll explain you the mechanism, how it works. Some scientific mechanism, I'll explain how it works. So deoxyglucose, it's very commonly used. It's called FDG, fluorodeoxyglucose. It's a chemical that's commonly used in PET scans, positron emission tomography, to detect the cancers. So that drug is used. So they have removed the fluorine and just used this sugar drug molecule to just test on the COVID patients, and it seems working. It seems like it's a couple of 100 patients were tested in somewhere in New Delhi hospitals, couple of hospitals. The study showed that some people are recovering better. What could be happening in the body is, so when the coronavirus infects our body, human body, so two things it does. One is it reproduces itself, makes its number more and more. Number two is it escapes. It sends some wrong signals to the immune system, and immune system produces lots of chemicals called cytokines and chemokines. Those are like bullets coming out of gun. They're coming to hit the virus, and virus escapes. They're going and hitting the lungs. So lung cells are being damaged, right? When lung cells are damaging, and then you cannot take the breath, or breathing issues come, problem. But the other side, what virus is doing is virus increasing the stress on the other cells to produce virus proteins, make virus more and more. So they're utilizing our own enzymes, our own body parts. So in our own energy to make virus more. So in energy, there are three processes. It's called when you eat the food, or the carbohydrates, or the food, or the sugars will be taken by the body and to produce the energy, right? Our cells produce the energy. So there are three processes. One is glycolysis, second process Krebs cycle, third is electron transport. These three steps happens in the body when we eat the food. That food processed through these three steps to generate the energy that we have. That energy is being hijacked by the virus and to produce its own proteins. So in these three steps, we don't need any oxygen. But in the third step, first two steps, we don't need oxygen. Third step, electron transport, we need a lot of oxygen. So one side, it's damaging the lungs. The other side, it's increasing the oxygen stress. So the patient is damaged two times. So the 2DG drug, what it does is, so it replaces the food. It replaces our carbohydrates, and then it stops these Krebs glycolysis step, stops the energy production at glycolysis. And you don't need more oxygen because you're not going towards the electron transport step, right? So that's how probably it's helping to the severely infected patients, and then they don't need more oxygen. They don't have to breathe more oxygen. They're recovering better. This is what it could be happening, but how well, we do not know. So it's not harming the patient. So if it is prescribed by the doctors, it's okay. You can take it. It's harmless. It's not, that it's used in the cancer cells because the cancer cells also utilize our own energy to produce cancer cells more and more. So that's why this drug only goes to the cells that are working more rapidly. Like, our cells are also like machines. It will not go to a different cells. It only goes to the cells which are using more and more energy. So that's how it probably. Yeah.
So then that way, can we say that it can be preferred over remdesivir then, because remdesivir excess use is leading to the black fungus or kind of thing, excess use of remdesivir.
No. Remdesivir, it's not. The black fungus is not because of remdesivir. Black fungus is because of inappropriate usage of steroids, unauthorized usage of steroids, unwarranted usage of steroids. Remdesivir is good for mild to moderate symptoms. The patients with mild to moderate, not the serious patients. Serious patients, even if you give the remdesivir, it's not going to help them, because the patient becomes serious because of the lung damage. It's not the virus anymore. The lungs are damaged. They destroyed lungs. Lungs need to be repaired. So probably 2DG may help to the lung repair.
So it's better over 2DG?
The steroids?
I mean, 2DG is better over
You mean
remedies?
not for the mild to moderate patients. I mean, if there is a lung damage, yes, probably 2DG may be helping. If there is no lung damage, I mean, I don't think the 2DG. So 2DG is not working directly against the virus. 2DG doesn't do anything to the virus. It probably indirectly helping our body to reduce oxygen stress.
Because
So 2DG is not.
serious patients, as you said, 2DG is also not working, because in serious patients, it's not working.
Oh, no, I didn't say that. I didn't say that.
No,
See,
you did
2DG, 2DG may. 2DG may be working on serious patients. Maybe I'm saying, it's only a couple of hundred patients were tested in the hospitals. It's not really like, very well studied. I'm not sure whether a lot of studies are ongoing yet, but
no, with our own, we heard, unfortunately, one of our senior retired professor's son, he was on serious note on coronavirus, and he was given 2DG, but sir, unfortunately didn't work out on him. So.
Oh, sorry to hear that. So again, that's what I'm saying. 2DG is not an antiviral agent. It doesn't do anything to the virus. 2DG is probably helping the patient to recover, reducing oxygen stress, reducing the oxygen stress on the patients. Probably that could be the possibility, but it's not an antiviral agent. It's not killing the virus. It's not removing the virus. It's not repairing the immune system. It's not producing the immune response.
Sir, thank you.
I hope I answered your question.
Yes, sir. Thank you.
There is another question: is why blood clots develop in COVID patients? It's a great question. So again, very rarely. So initially, majorly, the COVID-19 impacting the lungs only, right? But later on, as more studies are ongoing, it is also the virus particles also found in kidneys, in hearts, in liver, in various different organs of the body. So when it goes to the heart, it is doing something probably, don't know what it is doing to the heart. That's where probably this, even it's going to the brain. Some people are experiencing brain stroke as well. So not a lot of information known why the blood clots because of the infection. But virus is being damaging many organs in the body. Sir, after how many mutations virus will become weak finally? The Mr. Vadiraju is asking this question. There is no number, or you cannot say, because it's a virus consistently mutates. The time is what defines, it and the host. If you don't, the virus doesn't find a host, it will not mutate anymore. It will become more and weaker mutate. It will generate weaker mutations in a different hosts. If, let's say, healthy patients, the virus is mutating in healthy patients, the chances of going stronger mutations low. But the virus is mutating in cancer patients or immune compromised patients like HIV persons. The virus may become more and more stronger then, because it can easily mutate well, because immune system doesn't work well against the virus in people like immune compromised, patients like immune compromised. That's the possibility.
Dr. Mahendraji.
Sir.
Mr. Ravi is asking one question. In some people,
please go ahead, sir.
RT-PCR shows negative. However, their CT score is in double digit. RT-PCR is negative, but in CT scan, it is positive.
Yeah, there's one interesting, see, the CT scan cannot detect the virus.
Okay.
CT scan positive doesn't mean that there is a virus infection. CT scan positive means there is a pneumonia. It could be a pneumonia.
Okay.
the lung problem. There could be a lung problem. So there is no test. I mean, the CT scan test is not authorized to define the virus infection at all, anywhere in the world. That is that's a wasting of money and time. CT scans.
Okay.
Only RT-PCR is the one most effective. Sometimes, because of these variants, the virus can escape from the diagnosis from the RT-PCR as well. You know, in the beginning slide, I mentioned, right? That's why that's how classified variants of concern, variants of interest. Because variants of concern, sometimes they may escape RT-PCR test as well. It's very rare. It's very rare. But CT scan is not the test that to take to detect the virus. You cannot see the virus in the CT scan.
Okay.
Any scanning technology can see the virus. Only one microscopy technology, electron microscopy technology, you can see the virus. That's all. But those are probably not at all available in many places. I mean, that's expensive, very rare. So nobody would do it. Even like, for COVID, nobody would do it. So the RT-PCR test is the 95% efficient test for the COVID-19.
Okay. Thank you.
Sir.
Yeah, go ahead.
Sir, if RT-PCR is negative and the CT score is there, he should undergo isolation, sir?
So CT scan is positive. Is that what you mean? CT score is different from the CT scan.
Okay.
So RT-PCR test gives you the CT score.
Sir.
So CT scan is different. CT scan is different from the CT score. So there's the terminology is different. So if RT-PCR test is negative, the chances are very low that the person has infection.
Okay.
If it is negative, and if it doesn't have any symptoms, let's say if the person who tested with the RT-PCR negative and then he doesn't have any symptoms, then he's safe. He doesn't have to go for the quarantine or isolation. If he's showing some symptoms like, cold or, chest pain, breathing issues, that's not a good sign. So that means they need to go to get tested again and then go for the quarantine.
Okay. Thank you.
Yeah, you're welcome.
Thank you very much, sir.
Some administrator, I don't know who is administrator, asking some raised hand. Who is the administrator? You can unmute yourself and ask a question if you are.
Thank you very much.
All right, Sasija ma'am.
Many queries.
Yeah, go ahead.
Clearing many queries. And thanks a lot for spending so much time. Really, it is nearly.
This looks like one more question.
There's
one more question. Yeah, go ahead, ma'am. If you have any other questions, I do see.
Around 3 a.m. to you now. It is. The time is 3 a.m. I think. So you have been spent a lot of time. Thank you very much. And now please accept our participation certificate, sir.
Thank you. Yeah, there's one, Sasija ma'am, there's one question that last question I see I can answer that probably. It may not be. Just one question. Last is Mr. Nitin, I think he's asking. Dr. M.S. Reddy is a famous virologist, suggests to drink buttermilk daily to develop better immune system and explain that Indian food habits have potential to develop good immune system. I mean, it's, again, I mentioned also nutritional. How does that work? So, nutritional food is one of the component that strengthens our immune system. Not only the food, nutritional food could be, the buttermilk has the more probiotics in there. It's called more good bacteria that helps our body to recover better. I mean, helps our body to do our mechanisms better, work better. It could be, I do not know very much about that, but, eating healthy food, nutritional food, getting sufficient sleep, leading an active lifestyle, walking, running, that's very important to strengthen your immune system. Also, again, if you drink the buttermilk, today you drink the buttermilk, tomorrow morning your immune system will not become very strong. Also, today you eat nutritional food, and tomorrow morning your immune system will not be very strong. So it's a process. It's a weeks to months worth of process to develop a stronger immune system, and maintain it is very, very important. So always good food is important. I hope I answered your question. All right, yes, ma'am. I think sorry. Go ahead and you may.
I think we have taken lot of time from you. I think it's early morning of Sunday. Thank you very much, sir. Now I would like to request you to accept our appreciation certificate.
Thank you, ma'am. Thank you very much. Thanks for the invitation. Glad I was able to educate our people.
Thank you, sir.
Thank you.
Next, I would request Mr. Joseph to give vote of thanks.
Yeah, madam. Madam, am I audible, madam?
Yes.
Yeah. Good morning, all. On behalf of University College of Science, Satavahana University, I take this opportunity to propose vote of thanks to those who have directly and indirectly contributed to this international webinar on COVID-19 and expert analysis on variants, waves, and vaccines. At the outset, I thank our chief guest, Professor Mallesam sir, Honorable Vice Chancellor of Satavahana University, special guest, Professor T. Bharat sir, Registrar of Satavahana University, guest of honor, Dr. Vannal Ramesh sir, OSD 2 VC, Satavahana University, Dr. N.V. Sriranga Prasad sir, Controller of Examination, Satavahana University, and convener of this program, Dr. Sarsaja Madam, Principal, University College of Science, Satavahana University, and coordinators of this webinar. I would specially thank our guest speaker and resource person of this webinar, Dr. Mahendra sir, for your presence and enlightening us with your knowledge on COVID variants, waves, and vaccines. And you have given a clarification on efficiency of various drugs and vaccines, sir. Today's webinar gave deep insights into the topic and also revealed some interesting facts regarding COVID and vaccines and their variants. I personally thank Dr. Sarsaja Madam for organizing this program and organizers for conducting this webinar successfully. My heartfelt thanks to all the participants and our students for active participation. With these warm words, I would end my speech. Thank you, one and all. Thank you, madam.
Now we will join for national anthem.
Yes.
Vinayaka Bharata Bhagya Vidhata Punjab Sindhu Gujarat Maratha Dravida.
Thank you, one and all. The feedback form is available. Please click the feedback form for getting your certificate.
Okay. Thank you, Dr. Mahendra ji.
My pleasure, sir. Thank you so much. So I'm
Thank
leaving
you
the
very much.
meeting now.
I think I'm troubling you number of times. But
No problem, sir. Anytime, sir. I'm available. I mean, it's a pleasure actually. You're giving me an opportunity to serve our people. Thank you, sir.
But every time your talk is different, new, useful, informative. Really,
And sir,
it is highly
one thing I would
appreciable.
recommend, sir. I forwarded a video that I made about the vaccines in Telugu. Please forward it to a lot of people are asking in the chat window.
Yes.
Please forward it to that's particularly for the people from rural areas who have the vaccine hesitancy.
Yes, sure.
Okay, sir. Have a good day. Thank you so much.
Thank you so much.
I'm leaving there.
And convey my best regards to Madam and best wishes to your son. And thank you very much.
Thank you, sir.
I will talk to you personally after some time. Thank you so
very
much.
much, sir, Mahendra.
Sure, sir. Thank you.
For sparing lot of time.
My pleasure. My pleasure. My pleasure. Thank you.
Oh, congratulations, Madam Dr. Sarasija. I am Dr. Viraya. Hello.
Hello, Viray sir.
Congratulations, sir.
Thank you very much for your support and participating and motivating the people. Thank you very much.
It was a very good presentation by Mr. Ji.
Yes.
Hello, Viray sir.
Baunara madam.
Banan sir, Baunara
Very
sir.
happy. Link Vampichinanduku, I had this opportunity to participate in this conference. Thank you,
Sir. We thank you for spending your lot of time.
Thank you, madam. Once again, congratulations for conducting.
Thank you, sir. Thank you, sir.
Thank you, Dr. Mamata.
Sir, good afternoon, sir.
Very good afternoon. Thank you very much for participating.
Sir,
really...
very nice lecture, sir. Congratulations, Madam Sarasija, Madam.
Yeah.
Thank you, Mamta ma'am.
Sir, very nice, sir. So many number of doubts were clarified in this lecture.
Yes. Dr. Mamata, I am happy to share you that he was my own student.
Sir, nice, sir. Congratulations, sir. And we